Good morning. Morning, everyone. So today is the last day of the meeting with a lot of talks. And so today we start with Dr. Ganeshwaran Moshida. Dr. Moshida is a principal investigator in the Division of Genetics and Genomics at Boston Children's Hospital and assistant professor of pediatrics at Harvard Medical School. And he works with the molecular basis of genetic disorders of the cerebellum, brain development, and neuropathogenesis of Zika syndrome. And that's why he had the first connection here in Brazil. And today he will talk about the clinical and genetic overview of ATP1, AE3 related disorders. Please, Doctor.
Thank you, Luis. Thank you very much. I will make my presentation in English, but before that, first I'd like to give a few words of appreciation in Portuguese, so please bear with me. Bom dia a todas e todos. Sou Ganesh Mochida. E eu queria só aproveitar para agradecer aos organizadores, o Leandro, a Vanessa, a Jaqueline e outros, pelo convite. E também a Universidade Federal de São João del Rei. E também os patrocinadores por proporcionar esse ambiente bonito e muito acolhedor. É um prazer inenarrável estar aqui numa cidade histórica e conhecer vocês e as suas pesquisas muito empolgantes. Eu espero, sinceramente, que a nossa amizade, a camaradagem e o espírito de colaboração que nos vimos fortalecendo ao longo desse congresso dure muitos anos pela frente e também que nos avançamos essa área de pesquisa trabalhando juntos e, sobretudo, nos divertindo. Muito obrigado. So, let me get into my presentation. So it's a little bit different from the presentation that we have been hearing over the last several days because my background is pediatric neurology and human neurogenetics research. So I will try to make connection to some of the basic science talk about the ion pump. Specifically, I will talk about ATP1A3, many of you are familiar with, and its clinical and genetics aspects. Okay, so no conflict of interest. And so this is the agenda of my talk. I will give a brief introduction about ATP1A3 related disorders. Then we'll talk primarily about the most common clinical phenotype, which is alternating hemiplegia of childhood. I will use the abbreviation AHC throughout this presentation. And then I will only briefly talk about two other relatively common phenotypes, rapid onset dystonia Parkinsonism and Kaposi syndrome. And then I will talk about genetics of ATP1A3 related disorders, like, what kind of mutations do we see? And, whether those mutations tell us something about this protein and disease. And finally, I will only briefly touch on the new therapeutic approach that we are working on in collaboration with Dr. Al George, who's gonna, I believe, give a talk a little bit later. So I will sort of, give an introduction to his talk. So ATP1A3 related disorders, as probably all of you know, ATP1A3 is highly expressed in the neurons. So here's the ATP1A3, and then ATP1A2 is highly expressed in the glial cells. And so it is located near the synapse, primarily to restore the membrane potential after an action potential. The neuron excites, then this pump will restore the resting membrane potential. But as we have heard, there are many other functions, including the as a signaling molecule, for example. The important thing is that it's an autosomal dominant condition, meaning that out of the 2 copies of ATP1A3 genes we have, one from mother, one from father, only one has to be mutated to manifest the disease. So therefore, that's dominant. And then there are 3 major phenotypes, the phenotypes that I just mentioned: AHC, RDP, and Kapus. But it is important to recognize that they have significant overlap in symptoms. So AHC here, RDP here, Kapus here, and there are many symptoms that can be seen in any of these 3 conditions, but then each of these have some unique phenotypes. Therefore, they are, so they have different names. They are all devastating neurological disorders without effective treatment. I will briefly talk about the available treatment options. So first, I will talk about this AHC, alternating hemiplegia of childhood, because it's a very conspicuous presentation. So it is usually noted by the parents pretty early on. It's a rare neurological disorder characterized by recurring episodes of a loss of muscle tone on one side of the body. Actually, it can affect our whole body sometimes, but the most prominent feature is these episodes of hemiplegia. The majority are due to de novo mutation, so the parents do not have the mutation the child has in the ATP1A3 gene, although in rare cases, ATP1A2 mutations have been reported with this phenotype. So as I just showed in the previous slide, it's one of the clinical subtypes of the now what's called ATP1A3 related disorders. Many of the neurogenetic disorders that we work on had been known before gene sequencing became available. So many of them were renamed or grouped together after the genetic causes have been identified. So that's the same for this condition. And the prevalence is thought to be somewhere between 1 in 100,000 to 1 in a million. I have a sense, reviewing the literature, probably it is somewhat closer to 1 in 100,000 than a million. So that means that based on the Brazilian population, there has to be somewhere between 200 to 2,000 patients. And I believe, based on the cases we know through Leandro and our other colleagues in Brazil, this probably 200 is too low of an estimate. So it's a rare disease, but then there are substantial number of patients. Okay, so Okay, so how do you make a diagnosis? So here's like a clinical major diagnostic criteria here. And I will, you don't need to memorize these, but I just want to highlight a few important features. So one is that the onset of paroxysmal symptoms is usually before age 18 months. So it's very early. And then this contrasts with some other clinical subtypes that we're gonna look at. So this is important to remember. And then, repeated attacks of hemiplegia, which I will show you in video shortly, but then it could affect the whole body. There are also other paroxysmal symptoms, like, recurrent symptoms, such as dystonia, oculomotor abnormalities, like, abnormal eye movements, and autonomic symptoms, and so forth. The symptoms can be usually relieved upon sleep. But the other important thing is that there are also symptoms that are not periodic. So the child may have like permanent developmental delay, which we obviously would like to prevent if possible. So that's there are paroxysmal symptoms as well as more like static or non-paroxysmal symptoms. So I will show you the video of a child with a mutation cared at Boston Children's Hospital. And then, this video was given courtesy of the child's mother. And with her permission, I will showing you the hemiplegic episode. So look at the right arm. See, like, you can, the child cannot move the right arm much. And then the mom is holding.
Not really, huh?
And see, looks like it's very floppy. Even though he seemed to be grabbing using his left hand fine, and then sort of, right arm
I love you, strong boy.
hangs like this. So, and then, obviously the child is not very happy. In the, just want to make sure that I'm not skipping any slides. Okay, so there are triggers, known triggers to elicit these paroxysmal events. And then again, I'm not going to go through every one of them, but I just would like to highlight that like seizure disorders may have triggers. In like some of them, for example, illness, whether it's like viral or bacterial infection, so forth, sleep deprivation. Those are like fairly common triggers for seizures as well. Whatever the cause, generally sleep deprivation, acute illness, they can trigger seizures. But then some, others, they are sort of, more unique to AHC. For example, like, emotional excitement, environmental stress, such as like, light, heat, cold, water exposure. Yes, there are individuals who like, have seizures in response to these stimuli, but they are rare. But then in AHC, they are, these episodes are triggered by some of these stimuli. So I think, there is an important biological clue here. What's the pathogenesis? But we don't, fully understand. And now, seizures. So AHC patients can also have seizures. In fact, more than 50% of patients will have seizures during childhood. So not everyone, but then they do. The seizure types can be very variable. Only one part of the body or whole body, or patients may have mixed types of seizures. Status epilepticus, which is like continuous generalized seizure without regaining consciousness, is fairly common, and this can be very dangerous, as you can imagine. And then sometimes distinguish these seizures from other non-seizure paroxysmal events can be very difficult. So for seizures, commonly prescribed medications, including these relatively typical seizure medications. And then it's not entirely clear if there is anything different about seizures of these patients in terms of treatment response from the general group of patients with seizures. So I'm gonna show you the same child having a seizure. So it's a generalized seizure, so you can see the arms jerking, the eyes, fluttering with eye deviation. So it's a generalized tonic seizure and this child can have multiple seizures a day, which is very difficult for the parents, as you can imagine. There are also other severe symptoms, such as apnea, whether that can happen during sleep or during wakefulness. So, and then the oxygen level may also drop during episodes of neurological dysfunction. So, for example, this child has a pulse oximetry. If the oxygen level gets lower, the parents would give supplementary oxygen. And then in rare cases, there are abnormal cardiac conduction or arrhythmias. So oftentimes cardiac workup is indicated. So again, the same child having apnea during wakefulness, and then obviously he's given oxygen. It's hard to see if he's breathing or not, but then you can see that perhaps there is no sort of strong breathing effort during this brief video. So how do you treat these events? Actually, there are not many options. So during the event, obviously, first aid in any other emergencies, airway, breathing, circulation, and then oxygen as needed. So these episodes are generally relieved by sleep. So inducing sleep, whether just going to sleep naturally or using medications, have been sometimes tried. But then you want to prevent these episodes before they happen. So a lot of preventive or chronic treatments have been tried. Flunarizine, which is a calcium channel blocker, is probably one of the most commonly prescribed medications. In small-scale studies reported some efficacy in reducing the frequency and severity of the paroxysmal episodes, but then the effect is not optimal. Other seizure-related treatments, ketogenic diet, topiramate, have been reported in small case series as effective in reducing these attacks. They obviously have an advantage over treating the concomitant epilepsy if the child is having epilepsy as well, because these are both, effective treatment for many cases of epilepsy, and avoiding factors, circumstances that trigger these episodes that I showed you. But then, it can sort of impose a lot of restrictions in the child's life as well. So I just want to briefly mention about two other phenotypes. So up until now, I have focused on AHC. Here, I will just briefly talk in one slide about RDP and couples. So rapid onset dystonia Parkinsonism, the onset of dystonia and Parkinson disease happens very quickly, like within a few minutes, or may progress over a few weeks, but then stabilize, and then they may have some minimal improvement, but usually stay like that for the rest of life. The onset is most commonly in the second and third decades of life, so like adolescents, young adults, mostly common. So that's contrast to AHC. CAPOS is an acronym for cerebral ataxia, reflexia, pescavus, optic atrophy, sensorineural hearing loss. Characterized by early onset of cerebral ataxia with a relapsing course. So like several events of these ataxia, sudden loss of balance. And then, after each episode, then the condition like ataxia gets more and more like permanent. So in the onset is somewhere between AHC and RDP because the most common age is between 1 and 5 years. And also it is interesting to note that they have optic nerve involvement and sensory neural hearing loss, which we don't typically see in AHC or RDP. And certainly, these are really concerning, also, in addition to ataxia, concerning symptoms for the families. So now, going to the genetics part, so there have been more than like 200 disease-associated mutations reported in this gene. I think like more like 250 now. And then if you locate sort of these disease mutation on a 3D structural model, you can see that I will show you another scheme, but they are sort of clustered around the transmembrane domain. But you can also see these are all different phenotypes. So some of the mutations are associated with both AHC and RDP, or AHC and CAPOS. So there is a lot of overlap, even though there is some genotype-phenotype correlation. So this is more easy to see where the transmembrane domains are, and you can generally see these AHC-associated mutations, RDP-associated mutations, both seem to have a higher concentration in the transmembrane domain, although obviously there are some do not sort of fit near the transmembrane domain. If you look at the various phenotypes, so these are the various phenotypes, AHC here, CAPOS here, RDP here, and then number of patients reported between 2004 and 2021, AHC is by far the most common, more than 800 cases, although the next most common RDP is only like 140 something. So definitely AHC is by far the most common, and they are particularly commonly associated with these 3 mutations that we will see in more detail in the next slides. So 3 most common ATP1A3 mutations in AHC are these 3 mutations, accounting for about 60% of patients with AHC. So what it means is that if we can devise, say, treatment, customized like treatment to target these mutations, like these 3 mutations, we can possibly help the majority of patients with AHC. Pathogenesis of ATP1A3 related disorders is very puzzling and likely very complex, as probably many of you basic researchers in this field would agree. Many mutations have been shown to impair the ion pump function of ATP1A3, as we saw in some presentations. However, this does not fully explain the clinical presentations because the majority of pathogenic mutations are missense mutations. We actually don't know exactly how the complete loss of function of one allele of ATP1A3 in humans look like. We have some sporadic reports, but we don't know the sort of comprehensive clinical manifestation of these individuals. As we saw, there is a huge variety, not just the severity, but also how the disease manifests varies quite a bit depending on the mutation. So it's not just the difference in severity. And also the severity of loss of pump function does not seem to correlate necessarily with clinical severity. In this sort of statement comes mainly from the work by Kathy Swedner at MGH in Boston. So Dr. Swedner's group also proposed possible gain of function mechanisms because some mutant proteins may lead to abnormal protein folding or compete for the beta subunit, thereby exerting toxic effect. So based on this, Dr. R. George, our collaborator in Chicago, came up with the idea to knock down the mutant allele of ATP1A3 at the RNA level using ASO, antisense oligonucleotide, which is allele specific to the mutant allele. And then when this ASO molecule, which is complementary to the region involving the mutation, then this RNA-DNA heteroduplex will be attacked by RNase H. And then only this RNA containing the mutant will get degraded. So that's the idea. And then our group has been working with Dr. George's group to improve the efficacy and then improve the safety of the candidate ASO molecules that his lab has developed. So I just want to summarize what I discussed today. So ATP1A3 related disorders is a group of neurological disorders with variable but overlapping clinical presentations. AHC is the most common presentation. Available treatment that we saw is quite limited and not necessarily very effective. And we now, believe that there are at least some, toxic effects of these mutant proteins that we would like to get rid of. And then these gene-based therapies, there are many approaches that are being pursued, not just this ASO-based approach. Hopefully, they will bring hope for new disease-altering, not just symptomatic alleviation, but to alter the course of the disease treatments. So that is our goal. And through this work, I have been fortunate to work with various wonderful colleagues, both in the US and Brazil. So I work with my colleague in our division, Tim Yu, who has great expertise in developing these disease-modifying ASOs for like one or few patients, like individualized genetic therapy. So I'm working with his group as well as Al George and many other groups in the US, as well as in Brazil. So obviously the group of Leandro and Jacqueline. And it has been wonderful experience for me to work with these great groups of people, and then try to help these patients and family who are greatly suffering from these disorders. So thank you very much for your attention, and I'll be happy to answer any questions.
Thank you. Great talk. One question.
It's a great talk, Ganesh. Thanks. So am I correct in understanding that there are no truncations, nonsense mutations in the alpha subunit? Because you mentioned that there's no evidence that one allele knockout is seen in humans. That would suggest that there are no mutations that would cause premature truncations and degradation of the message.
Right. So, so it's a rather complex situation. One, there are hardly any like truncating mutations or truncating variants in the general population database, like NOMAD. There are large human databases. So that, usually tells that the truncating variant is bad.
Yes.
However, there are only few reports of this truncating variants in humans.
Right.
They had very mild phenotype.
Interesting.
But then they are like one of them is abstract and not even like a full paper.
Got
And
it.
also the mice heterozygous knockout shows much milder phenotype than knock-in of human mutations.
Got it. The reason I asked is because the ASO strategy then would work if it's a gain of function, because otherwise just removing the bad copy, and if the single remaining copy is not good enough, then you still have issues, but maybe milder.
So I think the sort of our general consensus is that the complete knockout of one copy is better than having a bad copy. But then whether that may not be enough. You're right. Either we may want to increase the production of wild type at the same time, but this is at least a start. Yeah.
Thank you. Very nice talk. So just following up on that and on the mechanisms, and it has been shown that some of the mutations alter the pumping function, right? And then intracellular sodium goes up, and the kinetics for sodium has been studied by, I don't know, several people, but like Vilsebente. So when you talk about a gaining function, I mean, what more is known about the mechanisms there in one case or the other?
So yes, I think so. For example, some mutations that are leading to misfolding, leading to ER stress and activation of ER stress proteins, and then leading to cell death. So the other purported mechanism is that the mutant protein, which doesn't have a normal function, may sequester the beta subunit. Again, still very early stage of understanding. But then I don't think this issue of intracellular signaling with these mutants have not been well studied. Even these mutant protein localization in neurons, I don't think have been studied, because I understand Dr. Swedner's group primarily used transfected ATP1A3 protein.
Yes.
So it's really I think something very interesting, and I believe Dr. George may talk about his IPS studies. So I think that is area exactly we'd like to know in parallel with this therapy development, because pathogenesis, therapy development, I think they go hand in hand.
And can I follow up and more from the, I guess, comparison with other neurological diseases, right? When you have, let's say, an increase in sodium, whatever, there is a kind of depolarization of the cells, and that will lead more with symptoms of the lack of tone in the muscles and things. But for the seizures, for example, what are the mechanisms for seizure, and how you can correlate these mutants with producing a seizure? Is it known?
No, I don't think so. So as I mentioned, it's not everyone who develops seizures.
Okay.
In the there are many seizure causing mutations in ion channels more than pumps.
Right.
So that's something very interesting yet puzzling. Some of the triggers are common to many seizure disorders, but some are not.
Okay.
So that's exactly something I would like to learn from the physiologists, pharmacologists, because as clinicians, it really we treat them, but not really knowing how they happen.
Thank you.
Thank you, Professor, who was a very nice talk. I'm very newbie about this clinical situation, and even with these mutations. These mutations bring some cell death in these patients, these neurons. How can you see about the effects of this cellular behavior and another cellular in the vizinhança, esqueci a palavra. Neighborhood, yes.
Yeah, yeah.
How this
That's
this protein mutate continue the cell degrade this protein or keeping the same way?
That's a very important question. So for AHC patients, actually, for any other phenotypes, usually they do not have any neuroimaging abnormality at the time of presentation. But over time, they may lose some brain tissue. And that's something that we definitely want to avoid. Couples, for example, you may see cerebellar atrophy. Atrophy meaning that, the shrinkage of the tissue. So clearly, over the long period of time, the cells get damaged and then they ended up dying. We don't know the mechanism, but then definitely, we'd like to use this early window of opportunity when the cells are still, there and then, sort of more or less functioning to preserve them. So that's what part of what I mean by this disease altering therapy. So very, very important point that I did not get into.
In the case of the protein, the cell tries to make some feedback behavior to solve the interaction of this ion pump with multiplication.
You mean when there is a mutation?
Molecular level, the protein, the cell tries to remove or overexpress the protein to compensate the.
Right. So we do not know yet, but we are very interested in that, obviously, because yes, the ideal situation would be 100% wild type protein and no mutant protein. So how those ratios we may be able to get some therapeutic effect with just a little bit of suppression of mutant protein, but we may not. So that those are all very important questions that we are trying to answer. So thank you very much.
Questions? No? Thank you very much.
Hello, my name's Steve Clapgoat from Leeds University. In the United Kingdom, and today I'm going to talk to you about
Okay, so now we have the presentation of Dr. Stephen Clapcoat. Dr. Stephen Clapcoat got his PhD in the University of Liverpool, and now he's a lecturer of pharmacology in the University of Leeds. So his research group focuses on the identification, characterization, validation of the drug targets for treatment of chronic central nervous system diseases. And today he's going to talk about these. Thank you very much, Dr. Clapcoat.
Hello, my name's Steve Clapgoat from Leeds University in the United Kingdom, and today I'm going to talk to you about a project that a PhD student of mine has been doing for the last few years, and the talk is entitled "Can Two Wrongs Make a Right? Rescue of Deficits in ATP1A3 D923Y mice by a second." Well, the effect of a secondary mutation in the sodium potassium ATPase alpha subunit was first reported by Bente Wilson's group at Aarhus University in Denmark. This was back in 2015. Rikke Holm in Bente's lab was investigating the functional effects of a rapid onset dystonia Parkinsonism and alternating hemiplegia of childhood mutation D923YN. That's the number. That's the numbering in the alpha 3 subunit. But she was investigating it in the alpha 1 subunit in Bente's lab. And during the course of that work, they realized that the clone that Rikke Holm was working on actually had a secondary mutation, and this secondary mutation in the alpha 1 numbering is E314D or E309D in alpha 3 numbering. And as you can see in this graph here, the mutant showed a deficit, the D928N mutant showed a deficit in the percentage of phosphorylation as the concentration of sodium was increased. Here in black is the alpha 1 wild type, and you can see here in violet there's a substantial rescue of that deficit in phosphorylation by the secondary mutation. Similarly, on the other side here, this is ATP-driven uptake of potassium in intact cells expressing either wild type or mutant sodium potassium ATPase. And as you can see here, there's a deficit in the D928N mutant compared to the wild type, but this is completely rescued by this secondary mutation in clones that had in cells that were expressing alpha 1 clones with both mutations in. So the sodium affinity of the alpha 1 D928N mutant was rescued by this unplanned second site mutation E314D. And after that, Bente then went on to check whether she saw the same effect in alpha 3. And in this case, they introduced a different mutation, D923Y, which is a mutation that is only found in AHC patients, not in RDP patients. And in this case, D923Y induced a substantial reduction in ATPase activity compared to the wild type alpha 3, but there was a substantial rescue of this, a twofold increase in activity compared with the D923Y mutant when the alpha 3 subunit also had the E309D secondary mutation. So this is maximal turnover rate for ATPase activity, and then on the other side we've got again, this is phosphorylation with increased intracellular sodium, and actually you can see here that with this measure there isn't much of a rescue, or as much of a rescue, perhaps a little bit there. So the sodium potassium ATPase activity of the alpha 3 D923Y mutant, which is in the M8 transmembrane helix, was rescued by the second site mutation E309D in the M4 transmembrane domain. And this is not a new thing. It's not new to see a second mutation having an effect on particularly a membrane protein that's involved in movement of ions. So another example is observed in cystic fibrosis. So in cystic fibrosis, a membrane protein called cystic fibrosis transmembrane conductance regulator, CFTR, this anion channel, is involved in movement of chloride across the cell membrane. However, in people with cystic fibrosis, they carry two mutant alleles of CFTR, two non-functional alleles of CFTR. So essentially, this CFTR can't work as a chloride channel, and the consequence of that is that there's a thickened mucus in the airways, and that causes multiple problems with breathing with people with cystic fibrosis. A deletion of a phenylalanine at position 508, or F508del, that is the most common cause of cystic fibrosis. And you can see where I got the idea for my talk from here. So this paper came out a few years ago in JBC in 2022. Can two wrongs make a right? F508del CFTR ion channel rescue by second site mutations in its transmembrane domains. So what these people did, this was from University College London. What these people did is they looked at the conductance, the CFTR conductance of wild type CFTR versus the mutant F508del CFTR, and what you can see here, there's a massive reduction in conductance. It's the mutant is essentially non-functional, and what they've done here is introduced mutations at particular sites to see whether these secondary mutations, in combination with the F508del, can increase conductance rates. And one of the secondary mutations increased conductance rates quite substantially. This was R1070W, which they identified as an effective revertant mutation, reverting the loss of function of the F508del. And they concluded that this partial restoration of function could potentially be achieved by base editing gene therapy. So base editing gene therapy in patients, or potentially in carriers as well, could potentially be used, whereby the arginine encoding codon is transformed to the tryptophan encoding codon. So this is another disease in which this second site mutation strategy is also being investigated. Several years ago, Bente Wilson asked me whether I would be willing to collaborate with her on testing an in vivo model of this second site mutation, and I was very happy to do so. So with funding from Bente's grants that she's got, we employed Cyogen, the company Cyogen, to make this mouse for us. And it's a conditional knock-in. So essentially the way it works is that two separate cDNA cassettes encompassing exon 7 to exon 23 are knocked into intron 6 of the endogenous mouse ATP1A3 gene. So the mice have exon 1, exon 2, exon 3, exon 4, exon 5, actually exon 6, and then they then express this first cDNA cassette that has the D93Y mutation in. They don't initially express the cDNA cassette with E309D in. They only do that in the presence of Cre or Cre recombinase. This first cassette is excised from the genome, and it's only this one remains. And the idea is that instead of this cassette with D93Y alone being expressed, we will now see expression of this cDNA cassette with both D93Y and the second site mutation E309D. And we've have done that. So this is an agarose gel, that the results of some PCRs that my PhD student Paria has done. So here we've got cDNA from brains from these mice, and this is genomic DNA from the mice. And these were primers designed so that they would amplify a band from the cDNA cassette, and would also amplify a larger band from the endogenous mouse gene. Because remember that these mice they're all heterozygous. So you can see here the wild types here with genomic DNA have only got this larger band, because they obviously don't express these cDNA cassettes, whereas the D93Y and the E309D mutants have both bands here. And then the cDNA, they're all expressing this lower band, which corresponds to cDNA. So far, we've only managed to look at total brain sodium potassium ATPase activity in the pre-Cre D923Y mice, but as expected, they do show a deficit in brain sodium potassium ATPase activity. This deficit here it's probably about 30% deficit, I'd say, which is smaller than what we've seen in the Mishkin mouse model, which actually has a severe neurological phenotype. These mice generally have a milder phenotype, and which is kind of consistent with the smaller reduction in brain sodium potassium ATPase activity that we see. We still need to get the results of the mice with E309D to see if brain sodium potassium ATPase activity is rescued by the secondary mutation. Now, the mutants do show a deficit in body weight. This is at 2 months of age. The deficit in body weight is noticeable from a younger age. And actually, in both males and females, that deficit is also observed in the mice with the E309D secondary mutation. It's not, so this is a phenotype which is not rescued by E309D, and that's seen both in females and male mice. Parrier has also conducted a series of behavioural tests on these mice. In the open field, the D923Y mutants show locomotor hyperactivity. So they walk a longer distance than their wild-type litter mates, and actually that's a phenotype that's not rescued by the E309D mutation. That those animals also seem to show locomotor hyperactivity. However, when we look at the percentage of time the mice spend near the wall of the open field, or at the periphery, what's called the time in the outer zone here, the mutants spend significantly more time in the outer zone compared to the wild types. But actually, this is a phenotype that is partially reversed by the secondary mutation. The E309D mice are still increased compared to the wild types, but they're significantly reduced compared to the D923Y mice. And actually, if we look at time in the inner zone, then that is more or less completely rescued by the E309D mutation. The D93Y mice spend significantly less time in the inner zone, but the E309D mice, they're pretty much at what? In another test, so in the open field, the increased amount of time spent by the wall is indicative of an anxiety-like phenotype. If mice spend more time around the wall and less in the center, it indicates that they may be more anxious. So the elevated plus maze was applied to the mice as a kind of secondary test of anxiety. And actually, opposite to what we might have expected. We might have expected if the D93Y mice were more anxious, they might have spent less time in the open arms. Well, actually, they spend more time in the open arms. So they spend more time in these open arms, elevated open arms, compared to wild-type mice. But that is a phenotype that is rescued in the E309D mice. Now, we were a little bit surprised about the result in the elevated plus maze. And so a colleague in Leeds had another piece of apparatus for testing anxiety-like behavior in mice, and that's this elevated minus maze. So Parrier tested the mice in that as well, and actually the results were pretty similar to what we saw in the elevated plus maze. So this is time in the outer zone, sort of around the edge, and what we find is that the D93Y mice spent more time in the outer zone compared to the wild types, but this was a phenotype that was rescued by the E309D mutation. So we think, on the basis of both the elevated plus maze and the elevated minus maze, it appears that the D93Y mutants show less anxiety, and that's rescued by the E309D mutation. In a test of hippocampal dependent memory, known as contextual fear conditioning, mice are put into a chamber with a grid floor. After a short amount of time, electric shock is applied to the grid floor and shocks the mice through their feet. And then 24 hours later, the mice are put back in the same chamber without a foot shock, and we measure the time that they spend freezing, because freezing is a kind of a defensive response of mice. So if they remember that, oh, this is the chamber in which I was shocked the day before, then they will tend to freeze. And what we see is that the D923Y mice show a deficit in freezing time. They freeze less than the wild type mice, and actually the E309D mice also freeze less as well. There's no rescue of this phenotype. So this would indicate a deficit in contextual fear conditioning. However, when we give a more challenging test of hippocampal dependent memory, this is called trace fear conditioning. So in trace fear conditioning, on the training day in chamber A, the mice, instead of just being given a foot shock, initially they're given a noise, a sound called the known as the conditioned stimulus. A sound is applied to the mice. There's then a 15-second gap interval before a 2-second foot shock is applied through the grid floor. The next day, we put the mice into a different chamber, chamber B, and we just play the tone. We play the noise. And if mice freeze, that suggested that they've made an association between the tone and the foot shock, even though they were 15 seconds apart. Now in this test, what we see, which is a more challenging test for the mice, we see that the D923Ys still show a deficit compared to the wild types, but in this case, with this particular hippocampus dependent memory test, there's a rescue by the E309D mutation. In terms of protein levels. So here we've got Western blots here. This is with an antibody from Santa Cruz. Paria actually saw an increase in protein abundance in the D923Y mice of the alpha-3 subunit compared to the wild types. That wasn't observed in the E309D mutants. We're not quite sure why this might be. However, that was not replicated with the alpha-1 subunit. With the alpha-1 subunit, we didn't see any difference between any of the genotypes. And the reason why we looked at the alpha subunit is that it's the other main subunit that's expressed in neurons. So to conclude, the D923Y mice have a 28% reduction in total sodium potassium ATPase activity in the brain. So that's made up of a combined total of alpha-1, alpha-2 and alpha-3. And the effect of the E309D mutation on this is still unknown. The D93Y mice exhibit reduced body weight, and there was no effect of the secondary mutation. The D93Y mice spend more time in the outer zone of the open field, and that was rescued by E309D. They also spend more time in the open arms of the elevated plus maze. Again, that was rescued by E309D. And they spend more time in the outer zone of the minus maze, and that was also rescued by E309D. D93Y mice show a deficit in contextual fear conditioning, but there was no effect of E309D on that phenotype. They also show a deficit in trace fear conditioning, but in this case, that was rescued by E309D. The D93Ys show increased expression of alpha-3 in the brain. That's apparently rescued by E309D, but we're investigating that further. And then finally, we still need to investigate by cDNA sequencing whether a transcript expressing both D93Y and E309D is actually expressed in the mice post-CRE. That work is still ongoing. And also, we still don't know if E309D rescues sodium potassium ATPase activity, and that work is ongoing in Bente Wilson's lab at Aarhus. With that, I'd just like to thank the people that did the work. My PhD student, Paria Anong Jaya, who's on a Thailand government PhD scholarship. Paul Armstrong, who was a postdoc in the lab, who helped Paria initially on her PhD project. And also I'd like to thank Bente Wilson of Aarhus University, who had this idea in the first place of a secondary mutation in the sodium potassium ATPase, who paid for the mouse model to get made, and who is also undertaking the sodium potassium ATPase assays of brain samples from my mice. And thank you very much for listening.
Thank you, Stephen. Do you hear me?
I hope you're still awake after all that.
Thank you. So it's open to questions.
Hi, Ganesh Mochida from Boston Children's Hospital. Thank you very much for the wonderful presentation. Some human ATP1E3 mutations have been associated with autism, for example. Looking at your behavioral studies, I wondered if, like, some of the changes that you saw in these mice, perhaps can be sort of, autism-like phenotype. Do you see any, have you looked at like a socialization of these mice?
No, we didn't do. One of the disadvantages of working in the UK is that that there are quite strong restrictions on animal testing, and the number of tests that we did, we kind of reached a limit on the number of tests. So unfortunately, we didn't do the social interaction test, which I think in an ideal world we would have done that on the mice. But in the other mutant that I've got, which is the Mishkin mouse, which we don't keep that one anymore because it was so severely affected that it was difficult to breed them and keep them alive. They definitely did have a deficit in social interaction.
Thank you.
Hi, Professor. Was a very nice talk. In the case of the secondary mutation that you did, how the structure behavior in the case of molecular interaction with the ions and mainly the van der Waals surface of this protein. This modification in the ion bind site is this?
Well, the thing about the two mutations, as I mentioned in the talk, they're on different parts of the in different parts of the protein. One is in the M4 and the other one is in M8, so they're quite distantly apart from each other. Bente Wilson has been doing structural modeling along with her collaborators in Japan. As far as what's actually happening, all I can do is actually just basically read out to you what Bente Wilson has said to me. If I can get this Word document to open on my computer. Let's try something else. Oh, sorry about this. Yeah, so in terms of the secondary mutation, she says E309D exerts a rescuing effect that is expected to markedly improve the rate of sodium and potassium transport under physiological conditions in the cell. This intriguing finding raises the prospect that a more efficient drug development strategy to treat ATP1A3 related encephalopathy emerges from using insight from the structural basis for this rescue effect. As the E309D mutation breaks a bond between helices M4 and M2 at the extracellular surface, breakage of this bond might be feasible with drugs administered via the blood, with specificity toward the M4 M2 interaction site. Yeah, that's as much as I know.
Thank you. More questions? So, Stephen, no more questions. Thank you very much.
Okay. Thank you very much for the invitation to speak, and sorry that I couldn't be there in person.
Thank you. Thank you. So now our third speaker is also here. It's Dr. Alfred George. He's the chair of the Department of Pharmacology and director of the Center of Pharmacogenomics in the Northwestern University in the United States. And his work focuses on genetic disorders caused by voltage-gated ion channel mutations that are responsible for disorders of membrane excitability, including diseases affecting muscles, heart, and brain. And so now his work is going to be presented here.
At the conference, and I'm really sorry. Hello, everyone. I'm Al George, professor and chairman of the Department of Pharmacology at the Northwestern University Feinberg School of Medicine in Chicago. I want to thank the organizers for the opportunity to present today at the conference, and I'm really sorry that I couldn't be with you there in person this week. I'm going to present our approach to studying ATP1A dysfunction in human neuron models of the disease alternating hemiplegia of childhood. The approach we use takes advantage of induced pluripotent stem cell technology, which takes advantage of the ability to reprogram any human cell into a pluripotent stem cell that then in turn can be used to differentiate into any cell in the body. In our case, we use this technology to generate cortical neurons from iPS cell-derived lines from patients that have defined mutations in ATP1A3. The table on the left in this slide displays the list of various ATP1A3 mutations that we've collected over the last few years and have reprogrammed into iPS cell lines. The first three mutations are well-known recurrent variants in this gene associated with alternating hemiplegia of childhood. I will be speaking today a lot about the third most common or most recurrent variant, G947R. From iPS cells, we can differentiate both excitatory cortical neurons and cortical inhibitory neurons. Illustrations of those neurons are shown here in these figures, where we've identified them using fluorescent reporters that are specific for each neuronal subtype. For today's talk, I'm going to focus on excitatory cortical neurons. These neurons are capable of expressing ATP1A3 protein levels that are detectable even in early developmental cultures. These are three images showing day 24 neurons, where we've stained for ATP1A3, or to collect the fluorescent protein reporter in these neurons, and then to show the merge of the two. As we approached this project, we wanted to determine what were the functional consequences in human neurons of AHC-associated ATP1A3 mutations. So we took a step back and thought about what are the main functional properties of the sodium-potassium ATPase, and what are the physiological consequences of its activity. Importantly, as I'm sure this audience knows, the sodium-potassium ATPase transports sodium and potassium with unequal stoichiometries, so that 3 sodium ions are removed for every cycle in exchange for 2 potassium ions. This charge imbalance results in a net outward movement of a single sodium cation. That generates a measurable but small current that's referred to as pump current. The activity of the sodium-potassium ATPase is necessary to create the sodium and potassium concentration gradients across the neuronal membrane that help establish the resting potential of the cell and also allow the driving force for generating neuronal action potentials. So this was the focus of our functional investigation. We used whole cell and cell-attached patch clamp recording of iPS cell-derived neurons from either healthy controls or patients with specific mutations, or patient cell lines for which we've converted the mutation back to the wild-type allele using genome editing to serve as an isogenic or same genetic background control line. And here are some of the things we measured. We measured pump current, which was a direct measurement of the ion transporting capabilities of the pump. We also measured potassium equilibrium potential, resting membrane potential, action potentials that we can evoke, and also the GABA reversal potential. And I'll show you sequentially why we measured those and how we think the pathophysiology of this disease can be related to these early and fundamental molecular events driven by the pump. The first challenge was to measure pump current. Pump current is the small outward current carried by the extra sodium ion conducted or transported by the pump. To do this, we first recorded current in the absence of extracellular potassium, a condition in which we would expect the pump to be inactive or at least have low basal activity. We then introduce a high concentration of extracellular potassium and measure the change in current that results. We then wash off the potassium, add in wabane to block the pump, and then repeat the potassium challenge. And the difference between these peaks we define as the wabane sensitive current. Doing this in a healthy control cell line or in neurons derived from an isogenic control of a patient line, we found this level of wabane sensitive current. We compared that to neurons made from cell lines that contain this recurrent mutation G947R. And there it was very obvious that there was a smaller amount of pump current generated with the initial potassium challenge. And quantifying that, we found that there was a statistically significant difference showing lower wabane sensitive pump current in cells with this ATP1A3 mutation. So this was, we believe, the first direct evidence that AHC-associated mutations in ATP1A3 impair the ion transporting capabilities of the pump. If the pump is not working to full capacity, then we would expect that the normal ionic gradients across the neuronal membrane that leads to low intracellular sodium but high intracellular potassium would be blunted. If that was the case, then we would expect that the amount of intracellular potassium accumulated through the action of the pump would also be blunted. Well, we have a method where we can indirectly measure intracellular potassium concentration. And that's done through this relationship between the equilibrium potential of potassium and its relationship to the potassium concentration gradient. And this formulation is referred to as the Nernst equation. The potassium equilibrium potential simply is the voltage across the membrane that equally opposes the driving force caused by the concentration of potassium across the membrane. We can directly measure potassium equilibrium potential, and in the case of healthy control neurons or isogenic control neurons, that value is, as you see here, around minus 90 millivolts, which is what one predicts from from normal physiological conditions. Because we know now the equilibrium potential for potassium, and we know the extracellular potassium concentration, which we control in the experiment, we can then solve for the intracellular potassium concentration. And again, when we do this in a normal healthy neuron, the calculated intracellular potassium is 127 plus or minus some millivolts, millimolar. And this is close to what is usually stated in textbooks as the intracellular potassium concentration of around 140 millimolar. Situation is very different for neurons with the ATP1A3 mutation. In that case, the equilibrium potential is very depolarized, on the order in this experiment of minus 64 millivolts. And that back calculates an intracellular potassium concentration of 46, vastly lower than the normal physiological level. We think this is really a critical physiological abnormality in these neurons. The reason we think that is because this is an important driver for potassium efflux through leak channels. Membranes of neurons have two varieties of leak channels. There are sodium leak channels, and by allowing sodium to slowly leak into the cell, that can depolarize the membrane potential. But that is partially and largely offset by potassium leak channels, which allow potassium to leak out of the cell down its favorable concentration gradient, and that has the effect of hyperpolarizing the membrane potential. And really, the resting membrane potential of cells is really governed by the leak channels in the cell membrane. Now, in the normal situation where you have a very steep potassium gradient, the leak of potassium drives the membrane potential toward the equilibrium potential of potassium, which is about minus 90 in neurons. In the case of AHC, where we have a blunted potassium gradient, and I just, for purposes of illustration, just said it would be around minus 60 some millimolar, the driving force for potassium is severely blunted. That's going to reduce the escape of potassium and have less of a driving force on the membrane potential, so there will be less degree of hyperpolarization. So from this, we would predict that the membrane potential at rest in AHC neurons will be partially depolarized. So we can directly measure that with patch clamp recording. In this case, we used cell attached patches because we didn't want to disturb the intracellular milieu and artificially affect the potassium gradient. So in this case, cell attached recordings of healthy neurons showed a resting potential of minus 59 millivolts. And as we predicted, the AHC neurons have a resting potential that's much more depolarized. In these experiments, around 8 to 10 millivolts more depolarized. So what are the physiological consequences of a depolarized resting potential? One consequence is on the availability of voltage-gated sodium channels. Voltage-gated sodium channels are essential for driving the generation and propagation of action potentials. So in an isogenic control neuron, we can record sodium currents, and at a artificially imposed resting potential of minus 80 millivolts, we get a very large inward current that's recognizable as a voltage-gated sodium channel spike. At near the resting potential of these neurons, if we keep the membrane potential there, we see there's a reduction in the amplitude of the sodium current, which was expected because some of those sodium channels are entering the inactivated state at this membrane potential. But there's still a sufficient number of channels that are available for activation to fire action potentials. That's in the control line. What about in the mutant cell line? Well, here, if we clamp the membrane potential to a level that is more aligned with the experiment, where we showed a resting potential of around minus 40 to 50, we can see that the sodium current amplitude is even more markedly depressed. And this might be sufficient to generate some action potentials, but there's likely to be failure of action potentials in some neurons because of an inadequate supply of activatable sodium channels. And this is what that would look like. If we looked at action potentials fired in neurons along an axis of different levels of stimulus, electrical stimulus, the control neurons show a rise and then a plateauing and then a fall in the number of action potentials fired in these neurons. And the effect of holding these neurons at the native resting potential, which is depolarized in the AHC neurons, we see less firing over the whole range of stimulus. And this is what that looks like. Here are action potentials fired in neurons from the isogenic control IPS cell lines. When we don't put any current in, there's no action potentials. But as we begin to scale up the current, we see more and more action potentials up to a point. If you really stimulate the cell hard, you get a burst of action potentials, but then there's failures. And this is due to what's referred to as depolarization block. There's insufficient time to recover the resting memory potential to allow sodium channels to recover from their inactivated state. And you get failure. That's expected. This is a normal behavior of a neuron. What about the AHC neurons? Again, when you input no current, there's no firing. When you put in a little current, you get firing. But then as you progressively add current, we get to the point where there's failure likely from depolarization block, even at lower stimulus input. So we think neurons with impaired ATP1A3 exhibit this greater susceptibility to depolarization block. What this ultimately means to a neuronal circuit will depend entirely on the balance between this effect on excitatory neurons and inhibitory neurons. And that's something we're still working on. Another byproduct of having an impaired sodium potassium pump and a lower than normal intracellular potassium concentration may relate to the maturation of GABAergic neurotransmission. And GABA is the most widely used inhibitory neurotransmitter in the brain. And although in mature neurons, GABA is inhibitory, it turns out that in developing or immature neurons, GABA is depolarizing or excitatory. And the reason for that is GABA acts on a receptor, which is a ligand-gated chloride channel. In immature neurons, the intracellular chloride concentration is high, and that translates into a very depolarized equilibrium potential for chloride. In those situations, if you add GABA, chloride is going to be driven out of the neuron in a direction that would depolarize the membrane. You're going to lose negative charges, and that's going to depolarize the membrane. Now, during development, several things happen, but one important thing is you get expression of this potassium chloride co-transporter protein. And this allows chloride to be transported out of the neurons alongside potassium. The driving force of this event is the potassium gradient. As this transporter works, you lower the intracellular chloride concentration. That then hyperpolarizes the equilibrium potential for chloride. And then when GABA acts on these receptors, chloride goes the other way. Chloride goes into the cell, adding more negative charge to the cell, and hyperpolarizes the membrane. And this is the mature neuron response to GABA, which results in inhibitory neurotransmission. Well, what happens in the setting of AHC? Well, these two neurons are redrawn here. Again, excitatory. GABA is excitatory in immature neurons because of high intracellular chloride. But when you activate the expression of the potassium chloride cotransporter, you lower intracellular chloride. And in the presence of GABA, chloride will move into the cell, and this will become a hyperpolarizing effect and an inhibitory effect in a network. Well, in AHC, rather than having a high intracellular potassium, we have a low intracellular potassium, and that's going to impair the driving force on potassium chloride cotransport. Therefore, chloride may remain high in a developing and even a mature AHC neuron. Therefore, GABA has unclear effects. So we tested that. We measured the reversal potential for GABA in immature neurons, and these are cultured iPS cell-derived neurons from a isogenic control. And at day 19, we would consider them immature, and the GABA threshold or the reversal potential is around minus 30 millivolts. So applying GABA in this setting is going to likely depolarize the neurons. If we wait many days, and these are day 83 cultures, these are really old cultures, applying GABA causes the reversal potential be around minus 70. And this is, again, more like a mature neuron. So we looked at the time course of this switch in the reversal potential over time in isogenic control neurons and neurons with the G947R mutation. And just look to the right here, this is the percentage of cells that have achieved the threshold which we assigned for the switch from immature to mature reversal potential at minus 60. The percentage of cells for the isogenic control rises and reaches a plateau around day 45 to 50. But in the mutation, there's a delay in starting the GABA switch, as it's called, and it doesn't peak completely, and it peaks much later. So we think this is evidence of impaired development of, or delayed maturation of GABAergic neurotransmission, which could affect the development of neurons and the maturation of neurons that could be a factor contributing to overall neurodevelopmental delay in patients with AHC. So to summarize the functional abnormalities we described, we have the primary failure or impairment of the primary transporting function of the sodium-potassium ATPase. This results in a less accumulation of intracellular potassium that blunts the potassium gradient, and that's going to have an effect on reducing the driving force for potassium to leave the cell through potassium leak channels, and that's going to result in less drive on the resting membrane potential, and we see this depolarizing effect on the resting potential. This in turn impairs sodium channel availability, which could affect and impair excitability of neurons and their ability to generate action potentials. And then lastly, it seems likely that this lower intracellular potassium concentration could also blunt potassium chloride cotransport, which will delay and maybe even impair the maturation of GABAergic transmission in the nervous system. So just some thoughts to summarize. In our experiments using this model system of human neurons derived from patient-derived iPS cells, we find that pathogenic variants in ATP1A3 can impair sodium pump function. I showed you evidence from one mutation, G947R. We have similar findings of impaired pump function from other AHC-associated mutations. We think this impaired pump function blunts the ion gradients. I focused this talk predominantly on potassium gradients. Almost certainly the sodium gradient may as well be very important. This affects neuronal excitability as a byproduct of depolarizing the resting potential, and we think it also contributes to delay of GABAergic transmission. So we think this is a good model system to test for the cellular dysfunction of ATP1A3 disorders, and it's also a good platform for testing possibly novel therapeutic interventions, such as small molecule activators of the ATP1A3 channel, something that we are very interested in studying in the near future. I give a thanks to the people in my laboratory, particularly Christine Simmons here in the front row, who is the person most involved in studying iPS cells in my lab. She's aided by Tatiana Abramovich. Also, Chris Thompson is the electrophysiologist who's been very instrumental in doing all the patch clamp recording. We have a collaborator at Northwestern who's helped us with some of the IPS cell technology, and some of the patient-derived lines originated at Vanderbilt University in collaboration with Kevin S., and also from Kathy Svoboda back when she was at Mass General Hospital. And we also appreciate our funding from the National Institutes of Health, and also the family foundations in the US that support research in AHC. I thank you for your attention, and I hope there's an opportunity for me to come on live to address questions you may have. Thank you again for the invitation, and I appreciate your attention today.
Thank you. Dr. George, do you hear me?
Yes, I do.
Okay, so questions?
Hi, this is Rajni Rao from Johns Hopkins University. Enjoyed your talk. It's actually a masterclass in neurophysiology, and I will be giving a lecture next week to grad students, and I'm going to try and incorporate some of what you've talked about today. But my question is, have you had the opportunity to look at the expression level of the alpha in these neurons, and whether they make it to the cell membrane, and whether they're still interacting with beta subunit and all of those? And I ask this because you mentioned that you are planning to look for small molecules to activate, but you might also want to correct for trafficking and folding and so on.
An excellent question. First of all, we have looked at the protein expression of ATP183 in the neurons derived from iPS cell lines with missense mutations. We don't see any difference in the overall or total protein level. We have not successfully done cell surface labeling of the pump, and so I can't answer the second part of the question, which is also very important. Is there abnormal trafficking? In terms of association with beta subunits, it's also something that I think is really critical to study. We've been sort of waiting in the background, watching and waiting for our colleague, Kathy Swedner at Mass General to work on this problem. We've been sharing some of our iPS cell-derived neurons with her, and that's a work in progress.
Wonderful, thanks.
Hi, Sandrine Pierre from Marshall University. Very nice talk. My question comes from somebody who has background with pump isoforms and other cell line differentiation from IPS, but not too much about neurons. So it may be a little bit innocent, but two questions. Manipulating isoforms in other model lead to inability to differentiate in some cell lineages. So in a way, I was wondering, listening to your talk, if there was something with the ability of the neuron itself to differentiate. So my first question would be, in the process of making those neurons, have you noticed anything in the markers of differentiation that was delayed or changed at any point of time? And that would lead to my second question, which has to do with the window of opportunity for reversibility.
Both are excellent questions. The main question about, is there a difference in markers of neuronal maturation in our cultures? First, I'd state that these are invariably neurons in culture that never really achieve full maturation. It's a well-known, accepted in the field of iPS cell-derived neurons that the neurons are always in some immature state. So maturation is a relative term. In our hands, we've mainly used electrophysiological readouts to gauge the level of maturation in culture. And so that's confounded, of course, when you have a mutation in the sodium-potassium pump, which could independently affect maturation. So it's a really, it's a chicken and the egg problem. We're not 100% confident that these mutations are affecting maturation independent of their impaired ion transporting activity. Your second question, please remind me. I did pay attention, but I can't remember now.
Yeah, that was, it's probably going to be difficult. In my expectation was that, well, maybe there is a window of opportunity through the process of maturation, but that would imply that you've been following those steps over time, and that's evidently a lot of work. But maybe that GABA switch, you can predict when this happen, and what would be a good time to try and address it, if any.
Yeah. Excellent question. And the question of reversibility, I think, is really important when we're starting to think about treatments of children with AHC. At what age has the damage unleashed by the disease been too great to reverse fully? And I think those are questions that we hope to try to answer in mouse models. And we have a mouse model in our lab that we've been working on for quite a few years. There are other published mouse models. I think the question of reversibility would be best answered there, because I think the reversibility may be postnatal, would attack events postnatally. And I think the cultured neurons may not be the best model for that. But it's an excellent question.
Thank you.
Thank you for very nice presentation. Natalia Fedosova, Aarhus University. I would like to ask a more philosophical question. Do you think that changing of the channels, sodium channels, or mutations in the sodium channels, voltage gated, or chloride channels involved in GABA transmission would be a better way to compensate for the problems with the sodium potassium ATPase? So instead of concentrate on sodium potassium ATPase, maybe we should try and modify the channels which will be adapted to a new concentrations of potassium and a new membrane potential. Thank you.
I think it's an excellent question. I think when we think of drug therapy in neurodevelopmental disorders, particularly those that have seizures as a comorbid phenomenon, the drugs that are used often block voltage-gated sodium channels or augment the activity of GABA receptors. And in both cases, those are strategies that are available now. I think that the general consensus in the AHC field is that sodium channel blockers have very low efficacy in treating seizures in that condition. But on the other hand, drugs that augment the activity of GABA receptors are really effective at seizure control, and sometimes have value in limiting or reducing the frequency of other paroxysmal neurological phenomena. And so I think you've asked a really important question: is the pump the best drug target, or should we be exploiting these alternative pathways? I think there's evidence now that the activating GABA receptors may have efficacy. It's just never been proven to be universally effective and fully effective. And so it may be that we still need some other strategies.
Yeah, but what I meant with sodium channels, I meant that they should be less sensitive to they should be activated faster. So you have to remove inactivating gate faster and not at high.
Yeah, no, I get your point. Yeah, so I think the idea is if there is a condition, as I've described, where physiologically sodium channels are not available because of the membrane potential, can we overcome that by removing inactivation? We have not tried that. There are concerns about this because there are many genetic disorders that affect sodium channel inactivation in the brain, and invariably cause very severe early onset epilepsy and epileptic encephalopathy. However, there is a new class of sodium channel targeted drugs that do partially impair inactivation, and these have been advertised as sodium channel agonists and may one day reach clinical trials for conditions where there's impaired sodium channel activity. So that it's not a far-fetched idea.
Thank you.
Oh, more questions? No? I like it, Joe. So thank you very much, Dr. George, and see you next time. Thank you.
Okay. Bye-bye.
Only God can introduce the Pope.
Yes. I'm really not sure if I have to introduce the Pope, but you know, anyway. So I have the pleasure here to introduce Dr. Leandro Barbosa. And he is Associate Professor of Biochemistry in the Federal University of São João del Rei. And as we all know, he is studying new synthetic cardiotonic steroids. But today, he will not present something about BD15, BD8, or BD1. He's going to present the iron effect on sodium potassium TPase activity. Thank you.
So thank you very much. You are kind of behind of schedule, so I cannot be fast for people not starving here. So Luis said very well. So right now we are full BD machinery in the lab doing all these BD in testing, but actually my PhD was in red blood cells. I worked in irradiation of red blood cells to see the effect on the sun department activity. So I like very much to talk about hematology and the pump activity. And then we start to talk about in the lab, like we try to find some pathological conditions that you can affect with the red blood cells to see the activity of the pump. In one of the conditions that we found in the blood banks is iron overload. So iron overload is a disease that you have increased iron concentration in blood in tissues. You can have a primary, that's hereditary, you have chain alterations related to iron homeostasis, and this you should show the name is hemochromatosis. And you can have a secondary iron overload, as a congenital diseases or acquired, people that have a lot of transfusion, a short period of time, you can get this accumulation of iron. Or you can have an increase of iron absorption with a progressive accumulation of iron. So iron is an essential element in our body, but the problem is if you have iron too much, you're gonna have a lot of oxidative stress. So how do our organism solve this problem? they put like a absorption of the iron very, very bad. So you can like, you have to do a lot of turnover of the iron in the body, and you have few very slow accumulation of iron through the absorption from the colon cancers, colon cells, enterocyte cells. So very briefly, you have like another problem that you have with the iron metabolism in your body. It's because you all have two states of iron, iron 3 and iron 2. And it's very easy transforming iron 2 and iron 3. So from the diet, you always get the iron 3, and for the entering the cells, you have to have a reductase protein to reduce the iron for iron 2, and this iron 2 can enter in a divalent metal transporter. So it's not, this transport is not exclusively for iron, not specific for iron. So we have a lot of competition for other ions. But when this iron enters the cells, this transporter needs proton, so this kind of a combination of NH3 transporter. And you see sodium here, so that's why driven us. So maybe the pump, sodium pump can be important for maintain this sodium gradient, for the have something to working here in this transporter too. So after iron 2 entering the cells, they have one protein that's ferroportin. This is a specific iron protein, only transport iron. So this iron 2 pass enter in the blood. But if you have accumulation of iron 2 in the blood, it's going to be a problem. So you solve this with another protein, that F18, that oxidate the iron, reduce the iron 3, oxidate iron 2 to iron 3. This iron 3 can make a combination with transferrin. That's how you can make the transport of the iron through the body. Also have some absorption of iron for other mechanisms. AIM proteins is the most effective, so that's why hematology has no AIM and the AIM iron. And you have other protein that I'm going to talk about. It's lactoferrin. That's a protein that you found in milk that transports iron 3. And to have a specific receptor for lactoferrin, and then you can have the uptake of the protein, then associate the iron, and then have the absorption of the iron for this protein. So iron overload or hemochromatosis is a toxic condition for living cells. You see the effect is a very slow accumulation, and the symptoms appear in 30, 50 years old. So have this problem because have a lot of lipid peroxidation, fibrosis, host production, DNA mutations, have some association with diabetes mellitus. And the major problem is going to be in the liver, because it's where the storage is found. And again, you have this iron damage because you have a fatal reaction during the iron 2 to iron 3. And this hydroxyl radical that's very, very, you don't have enzymes to destroy that. So this hydroxyl can virtually destroy everything. DNA, membrane damage, protein damage with carbonylation, influx of this calcium and sodium, water, and the mitochondrial damage. And you don't have any treatment for that. So if you have iron overload, hemochromatosis, the only thing that you can do is a regular phlebotomy. So this patient was always checking the levels of transferrin ferritin in the blood, and then start to become high. They go into the blood bank, have some phlebotomy, and that's it. So this was an opportunity, because this blood they throw away. So I started to think, look, maybe this is going to be very good. You have a lot of samples. Get good blood from blood bank is very difficult, because they don't want you to donate for research. But this is like, for them, it's garbage. So let's try to do this. And when we started to think about, at the beginning, I have a master PhD student that developed this project. We thought, look, it's going to be almost with the irradiation. Going to be a lot of ROS. Probably this ROS going to damage the sodium potassium ATPase. You're going to have an inhibition of the pump. So let's do it. We're going to be perfect. But we found some results that maybe the pump itself is going to be important since they control the gradient of sodium for the uptake of the iron for the cells. So this is a paper that's showing that they calculate the accumulation of iron, the uptake in the cells in the bronchial epithelial cells. And you see that when he uses wobble, they block the uptake of iron. So in fact, they did several auto transport that cause kind of the same inhibition, but for the pump, it's going to be important because they are several of those are related with the gradient of sodium needed. So we start to think about, okay, maybe it's going to be interesting to see how pump is going to be causing or related with this accumulation. And another thing that's interesting, when you check the papers, several papers in the literature, you always see inhibition of the pump, as we thought at the beginning. But something that is very interesting, because the majority of the papers that you have with iron, to check the sodium potassium base activity, is iron 2, not iron 3. So we have luck, because at the time we don't have iron 2 in the lab. So just to use it, let's try to see how iron 3 can contribute. So we have kind of 3 parts of the lecture today. One's going to see how the iron 3 the patients with hemochromatosis, how it's going to be the activity of the pump. Then we tested the in vitro with the iron 3, and after we tested the lactoferrin effect to see if it's a protein associated with iron 3 can have some effect too. So first of all, collect the samples in the lab. So poor Marco. So the in vitro is very difficult because you need some subject in the lab for that. Marco was the best subject because his activity of the pump is so high. So sometimes you see error bars. It's because of Marco's blood. So what we started doing at first was start to get these patients. Here I have the study that we published in 2015. You have 14 patients with hemochromatosis. And as you can see, the first thing that you try to see is like, okay, let's try to see the effect of lipid peroxidation in the damage, ROS production. You can see that hemochromatosis patients has more lipid peroxidation. And to try to mimic this in vitro with a condition with hydrogen peroxide, 1 micromole of iron, and you didn't see so much this effect in vitro. This we tested the normal blood cells, red blood cells, erythrocytes for 24 hours, okay? But even though you see that you have a decrease of cholesterol and phospholipid in the patients, maybe because of the have ROS increased, but this is kind of interesting because the iron, even that they don't cause lipid peroxidation, was able to cause mimic the same effect that you found in the patients. So probably iron can modulate the amount of cholesterol and phospholipids on those cells. And for our surprise, you see this rush activation of the sodium potassium base activity in the patients. And you was able to mimic that with the treatment, 24 hours treatment with iron. So, and even that, just to see if it's not only because of the ROS production, the ROS that we added did not have the same effect. So probably we are, okay, maybe it's the iron itself that has the effect over the pump. One thing that you try to see, just to be clear, is like, if it's the iron, the amount of iron, or maybe some protein that transports iron that make association of the pump. Because I have some reports in literature that ferritin can do some activation or inhibition of the pumping MHC cells. So we took the blood and to remove all the proteins, you wash the cells and just make it saline with the iron, and the effect continues the same. So this is a clear indication that it's the iron itself that is doing the effect in the pump. So to understand more, we're starting to see, okay, let's make in vitro, because you just treated with 1 macromolar. We read some papers to see that maybe it was kind of high concentration of iron. And you can see here that you have a biphasic effect that at the low concentration until 1 macromolar, that is the maximum that you found. You see this huge activation of the pump. Then when you start to get more iron, you start to see the inhibition. It's important to have the iron 3, probably is important to check that you treat it with TDT to see that, so you're gonna reduce the iron 3 and to have a blocked effect. So probably this is iron 3 effect, not iron 2. And even that it was iron 3 for iron 2 is completely different element. How the amino acid can attack, everything is so different. And to be clear of that, we tested with iron 2 and you see no activation of the pump. So this confirms our first hypothesis that iron 3 is the element that's important for making some association with the pump. So the question that the answer after was, okay, I have this effect on the pump. So, but what condition this iron maybe can have this interaction? So, as you know about the cycling of the pump, you can driven the pump for a 1 state or a 2 state if you're using more sodium, more potassium. So, starting to doing this, and start to make a lot of pre-incubation with iron and with sodium or with potassium or magnesium to see if maybe with different conformation have the same effect. And it's interesting, like if you see here with E1 conformation, still with 1 macromolar that you have the activity, you didn't lose the activation, but it maintains the activity. But look for if you have in E2 state, you have only inhibition. And with magnesium, you almost blocked the effect of the iron. So we think that here is an indication that maybe it's more associated with V1. And here maybe the iron maybe have to do some complex with ATP to coordinate. The college showed that there's some papers that show that iron can make a complex with ATP. So maybe when we pre-incubate with magnesium, magnesium was more competitive to bind with ATP, not doing this complex, and you block the effect. Again? So the problem with doing this in red blood cells is because red blood cells don't have DNA, don't have synthesis. So how can it be this effect in a living cells that has all the machinery? And so for that, you use an enterocyte cell that is important for the absorption of the iron. And you see the same effect, but they have like a left shift effect with less concentration of iron. We still continue to see this effect. But look, that's something that's important. It's interesting. When you take the cell membrane preparation of these cells that only have alpha-1, you don't see an effect of the iron. So we started to think about that maybe this effect can be like isoform, some kind of isoform selective, because red blood cell has alpha-1 and has a little of alpha-3, and not alpha-2. So for confirming that, you get the red membrane preparation test with the iron, and when you see the organ sensitivity, you see that the alpha-1, you didn't have effect, but again, the alpha-2, 3 fraction, you see this activation. So since the red blood cells don't have alpha-2, we believe that this is some effect of alpha-3. And to confirm that, we did some kinetic curves in ghost erythrocytes. And again, you see like the potassium curves, you don't see nothing, not so much difference, but look for the sodium curves. It's the activation is there, and look how much difference it is. And just to confirm that this is kind of isoform specific, when you do this in the purified kidney enzyme, you didn't see the same effect. And when you try to see this in the kinetic parameters, you can see this. Look, the potassium, not change too much. The kidney protein, not so much, but look at the sodium kinetics. It's almost threefold affinity for the iron covers in the sodium. So altogether, we believe that this data may suggest that the E1 subconformation of the enzyme are required for iron modulation, and that an isoform other than alpha-1 might be involved in this process. So, but what about it's a protein associated with iron can have the same effect? So this is a fair question, because you see that the iron is kind of the transport of iron in the blood is through proteins. So using lactoferrin that transports iron 3 to see if you have the same effect. For our luck, you get the same effect to see the activation of the lactoferrin. This is in the red blood cells, viable red blood cells for 24 hours. You see that you have this activity of the pumping. And here in the membrane, after 30 minutes, you see the same effect. In very low concentrations, you got this activation. And again, when you test this in the head-brain membrane preparation, you see the same pattern. In the alpha-1, you didn't see effect. For the alpha-2, 3 in the brain membrane preparation, you have the same effect. So probably what you think about that, maybe we think about like, okay, but this is an interaction of the lactoferrin protein together with the pump, or this is coordinate because they have the binding side of the iron in the lactoferrin. This iron kind of driving the conformation for the interaction. So we just did like a PMAL interaction with the lactoferrin that you have in the data bank with the alpha-3 and the alpha-1. And you see here in yellow is the sites that you have the interaction. You can see, look at how interesting, like, have so much interaction with alpha-3 compared with alpha-1. And the most important, when you wrote the paper, we start to think about, okay, what gonna be the sites of the alpha-3 or the pump that can maybe have interaction with the iron 3. So we think about it that maybe the phosphorylation domains here can have some kind of, gonna be like good candidates for have interaction with the iron 3, right? And when you see this interaction here, you have the same, almost the same interaction we found with the lactoferrin in the alpha protein. But something that's very interesting, those sites has like a very, for the alpha-1 residues, it's more than 20 angstrom of distance. So it's not very good to have interaction, not strong, compared to alpha-3 that's less than 20 angstrom interaction. So probably it's because of that, but this is just a preliminary, this idea, that you have more alpha-3 interaction with iron than with the alpha-1. And these sites, that is because these lactoferrin they are crystallized with lanthanum, and this is the interaction that you have with the ion lanthanum of the lactoferrin. So not only non-specific protein-protein interaction, it's interaction between the ionic that's there, the iron that's there. So we believe that this is very interesting because how the importance, how can be the interaction of the iron with these isoforms, and how it's opening up several questions, like with a patient that has hemochromatosis, how these can affect other properties that is driven for the sodium potassium activity. So in summary, you have patients with iron overload have increased their erythrocyte sodium potassium activity. Activation effect is dependent at iron 3 status, so it's not the iron 2. The iron effect appears to be dependent of a E1 conformation of the enzyme. Iron 3 effect appears to be dependent of 3 isoforms. Lactoferrin, that's transport of iron 3, causes similar effect of activation of salt potassium activity. That's it, you see with iron alone. And you are confident that the lactoferrin modulation is probably coordinated by iron 3 interaction with the sodium pump. Another thing that we cannot, we don't know the importance. It's another, how say, like thought to have. Alpha 3 specifically, but maybe gamma can be important too. You have gamma in red blood cells, but very low amount of gamma. But the kidney have a lot. So you don't know, like, if the gamma, if the amount of gamma can change the binding sites or the association of the iron 3 with the sodium pump. So have a lot of stuff to confirm. So it was quickly. So I just want to thank all people in the lab that we always have the best parts in my university, 2 per year. So people are always happy. And I want to thank the professors that work with me, share the lab, Vanessa, the Queen, Erica, Luciana, Drummond, Graziele Maia, that's lab manager, that's put all together the labs. Without her, it's going to be no possible work in the lab. Dr. Luis Quintas, that's help us with the kinetics curve. Same with my former mentor, Carlos Frederico Leite Fontes, both from UFRJ. And I want to thank the 2 students that did the project. Tamara Fernandes Costa, she was in NIH for a while after she finished the PhD. And Lelismara Souza, she did the hemochromatosis patients working, and then the lactoferrin work. And now she's a professor of hematology in the Federal University. So thank you very much.
Thank you, Leandro, for the great talk. Questions?
So I'll repeat, very interesting. It's really a very interesting scope of the work and new. So I maybe missed something. I saw that you were demonstrating that it does not depend in any protein in the plasma or in the blood, because you separate.
Yes.
And then you're demonstrating that this protein, lactoferrin, is the one that may be interacting. So did you lose this lactoferrin in your separation, or what happened?
No, it's kind of, it's not exclusive. The first of all, that when I wash, is I want to see if the iron alone, just by itself, can make the interaction with the pump and cause the activation. So when I wash everything, it's okay. It causes the activation, so you have the iron affected by itself. The second question is, okay, even that the iron alone do that, if you have a protein that transports iron, can this protein by itself can have the same effect?
Okay.
And to prove the lactoferrin.
Okay, now it's clear. Thank you.
But we didn't test with the other proteins as well. Something that I wanted to test: ferritin, not lactoferrin, but transferrin, if it has the same effect or not.
Okay, thanks.
Hi, Leandro. I think you're essentially mentioning this in terms of how the iron gets in. It's presumably bound to protein, but you can imagine the form of iron that you treat could have a big effect, right?
Yes.
In terms of its ability to get incorporated by the protein. So you're just starting with iron chloride, is that right?
Yes.
And so what about other iron sources? Have you tried that?
No, never. Because this is a good question. You have a lot. The free iron in the blood probably is bound with citrate.
You think it's citrate?
Yes.
That's
you
what
have.
I was about to suggest.
Yeah,
Citrate.
so like Tamara, that was the student that make all the kinetic stuff. During the PhD, her plan was treated the mouse with the iron citrate to see if you have some effect. And because you're gonna have more tissues to collect and see the effect, not just blood. The problem is that like when she entered the PhD, we have no fellowship for her. And then she moved. And we was not able to do. But I agree with you, like you have other status of the iron that you have to test to see.
So you're familiar with Popeye, the sailor man?
Yeah.
Okay. So he eats spinach, and spinach is a source of iron and strength. But then we learned later that because spinach has a lot of oxalic acid, then apparently it doesn't get absorbed. And so that's what made things that citrate could be a non-absorbing form of iron. And so I would iron citrate, but I would think iron sulfide. There's lots of just, you'd want to have like a standard experiment and think about different irons.
Yes, iron sulfate, you tested, didn't have effect.
And so I imagine that protect the sulfide because it already has the sulfide, so it doesn't get to the protein.
Yes.
But I imagine you could imagine, I think be creative. So I think citrate, but also then iron oxalate and then others.
Yes, absolutely true.
So I used to work with red cell ghosts in 1975, and I never expected to hear anybody talking about them again.
I know, like my first pump meeting in the Woods Hole, I was with my PhD process.
Yeah.
It appears in my poster because it has blood cells, right? But I was not able to explain for him because my mentor just popped me out for the poster. No, I want you to talk in poster, not you. But I like the model. The problem is you have some limitations because you don't doing the Western blot part, the alpha 1 in the head blood cells is almost impossible. Try several times, so low amount.
Yeah, really difficult. So that was one of the things I was going to ask you about. Back when I was using them, everybody thought red cells only had alpha 1. And then you mentioned
Hoffman has
there's a little bit about alpha 3. So what's the relative proportion of the two isoforms, roughly?
I don't know. It's very low. It's not. This is something that
is it 10% or?
They only have Western blotting. They did because he did immunoprecipitation. No, not immunoprecipitation. He did, I think, a column with the antibodies and then eluted to see. But it was like the alpha 3 maybe 30-40% maybe.
Oh.
I think 30%, something like that. The gamma is like just a trace by the head. They found
okay. So the other aspect of working with ghosts is you get ghosts that are unsealed, and then you get ghosts that are sealed right side out and ghosts that are sealed inside out. So there's usually a mix. So the question would be, could the effect of iron in the ghost be due to unsealing and allowing magnesium ATP to get to the catalytic site?
This is a very good question. I didn't have this yet.
You don't know that, yeah.
No.
So there's a way to make ghosts so they're all unsealed,
yeah,
or they're all right side out or inside out.
You know what the problem is, John? It's like people to do the ghost is so work. You have to wash. The students want to do like BD, so it's difficult to find someone. Look, let's move it to the head blood cell again.
But you have a good donor.
Yes.
I mean,
it's
you
true.
might as well bleed. The Pope may as well bleed his population as best he can. And the other thing is, do you actually do the venipuncture, or do you get somebody?
I did.
You do it.
Yes, I worked in a blood bank for 2 years. So.
Oh, I see. So the Pope became a vampire.
Yes.
So, Leandro, when I was a student, there were always some old professor sitting in the front row who would refer to some experiment 20, 30 years ago and say, did you check for that? And now I am that professor. Okay, so I remember that there was Steve Carlish, like 20 years ago, showed that iron cleave the sodium potassium ATPase. And I also remember from other proton pumps, right? It's concentration dependent, so that at low concentrations, the first clips actually activate the pump. They work a little faster. And then, of course, as there's more proteolysis, then you see an inhibition. So my question is, have you looked to see on running these on a gel after iron treatment, if there's any proteolysis?
I never did, but it is a very good question. Like, I remember the problem that the only thing that is different from the college paper was great for me, explain a lot of the results. But he used iron too, iron sulfate. So maybe because of this, we believe that it's going to be a kind of different mechanism. But it's, yes.
Very nice, Leandro. So these patients with hemochromatosis has more activity of the pump.
Yes.
So is that giving the cells an advantage? Is this just a phenomenon that goes, or is a way to compensate and try to maybe get rid of I don't know.
Yes.
I mean,
At the first, remember that I presented the first part of this history in the SPEFT. So since the activity of the pump is important for the iron uptake into the cells,
right?
So maybe what I thought is like, okay, you're gonna have compensation, increase the activity of the pump, and to remove the iron from the blood into the cells. It may be gonna be a modulatory effect.
Yeah.
But at the same time, if you have this in the enterocyte, you can increase the activity of the proper NH3. And you can work more the divalent transporter.
Right.
So
yeah.
So it's difficult.
Right.
One thing that's going to be very, very interesting, very interesting. If I got one like all enterocyte cells or a liver cells with pump activity decreased, like a knockout of the pump.
Right.
To see how gonna be the uptake, and how, because the problem is iron into the cells has deal with a lot of stuff. Have ferroptosis, have iron protein related to iron that has translational more. So have a lot of going on, but you have to look not so much for the pump, to look more for the iron metabolism. It's for that to have need a system that has nucleus and has expression of the protein to check.
So we may have the model for testing that. So
Maybe this will be BD15.
To see how it's working. This is nice. Another thing that is difficult, like I found, there's one group that tested how you have the uptake of iron in the cells is very difficult. It's not an easy task. So they use radio labeled, so that's impossible for me. So this is another thing that's going to be interesting for the Keiko2 cells. I use you have a dye that you can see this. If you make a slide, you can see with more iron, see this effect. And when you see in the enterocyte cells that you found this effect, the uptake for increasing the iron inside the cells was very, very high concentration. So at that concentration that they use is 0.001, you don't have so much iron inside of the cells. So maybe start to make the compensation to modulate the entropy, and it to have so much, you cannot block anything. So I have a lot of questions yet.
Thank you.
Hi, Professor. Is about the how hard is working with these red blood cells? You are saying this is difficult because of lack of resource to work with a lot amounts of these cells. But have you think about working with the precursor of these red blood cells? Maybe immortalize. My hematology is not good. I think it's megakaryocytes.
Yes, this is important too, because I remember that one of the reviewers asked, look, if you see the expression of the alpha-1 in the reticulocytes, it's different from reticulocytes because we have remodeling of the plasma membrane because they get out of some part of the nucleus that they still remain. So the guys thought, look, do you think that this activity that you have in the patients is because you have increased in reticulocytes and that led some more? But when testing the vitro, you see the effect. So, but I agree with you. Like, how can I be the effect on younger, immature erythrocytes lineage? We don't know, and maybe this is going to be a problem for the maturation of the erythrocytes.
Just to tell you that in our group, in our department, there is a professor that works with
Drosophila.
Drosophila.
He reviewed my paper.
Yes.
Yeah.
And he measures every metal you want. So you can make the animal that you wish to. And then you have the organs and you can lyophilize them and see if there is a storage in the
Exactly.
storage has changed during this process.
Yes.
Or in these animals that are a model for your disease, of course.
It's a good idea.
More questions? No? So thank you very much, Leandro.
Thank you.
So now we have a lunch break and we come back at 1:30. That's it? Yes? 1:30. Thank you. We can begin now. You all to be there in the next year. The 7th International Conference of P-type ATPases is in Health and Disease. This is a very traditional conference that will be held in Cairns in Australia in September. So we have many topics there. Sandrine is in the committee, organizing committee, me too. So we want to invite you. I know it's a long distance from Brazil, US to Australia, but it will be cool. So thank you all. We are going to present the next talk. I want to introduce Did you took a picture, Sandrine? 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 Did you took a picture? Did you take a picture of that? So just no. So now I have the pleasure to introduce Dr. Pedro Azalim. Pedro Azalim did his PhD in pharmacology and medicinal chemistry in Federal University of Rio de Janeiro, and now he's a postdoc in our lab there in Rio de Janeiro. And he studies about cardenolides and bufadienolides and the structure activity relationship of them. Today he is going to present drug target kinetics and thermodynamics in drug discovery using PNPP to characterize the ligands of sodium pump in a cheap way. Thank you.
So first of all, I want to thank everyone involved in this beautiful meeting. I want to thank you, Leandro, for allowing us to show a little about our project that we are working. So our main focus in the lab is about the characterization of this cardiotonic steroid, and we are in this moment try to characterize the thermodynamic that connects the behavior of this interaction. So we have been working with a lot of cardiotonic steroids. So we also always keeping the cis and cis configuration of the steroid core, and we have working both with the cardenolides and bufadienolide classes. And we have changes. We have many modification of this steroid core in the case of number of hydroxyls in the core. Also checking the cyclization that can show up in some bufadienolides. And of course, we working with the different oxidic portion, like 3-digit oxose or even just one aminosis. We have also worked with Professor George in the case of the modification of oxidic portion. So in this case, we have always working with the steroid core of disoxygenin that's more hydrophobic, and changing the and bring modification of this oxidic portion by synthesization by de novo substitution, by de novo synthesis asymmetry of the carbohydrates to bring the steroid oligoisomers of aminosyl and aminocetosils. So the important thing about the interaction of this protein and characterize the ligand is this protein perform a lot of work to complete the ATPase cycle. So this protein change for many subconformation of A1 conformation that allows the binding of sodium and perform ATP hydrolysis, and a lot of subconformation of E2 conformation that allows the binding of potassium and start the phosphatase activity. And it's well known in literature that the cardiololides bind with more affinity to H2P conformation, that the binding site is more open to this cardiotonic steroid. However, it's very important to notify that this is not true for all cardiololides, all cardiotonic steroids. So, as Laus and co-workers have shown in the group of the Professor Fedosova, they show by showing the residual activity over different concentrations of the steroid cardiotonics. We have seen that the digitoxygenine, when you increase the concentration of potassium, the affinity is reduced. However, when you take a look for bufalin, this is not happen. And sometimes, depends on the situation, some can be increase of the affinity. So later, what the Kanai and co-workers have shown that this alkadenolides, in the case exemplified by all cardiotonic steroids, in the case exemplified by bufalin, they interact indirectly with the potassium by the water molecule. However, when the potassium is present in the ion bind site 2, the bufadienolides characterized by the 6-membered lactone ring interact directly with the potassium in the bind site 2, which does not happen for cardiololide in the case of bufalin, wobain. In the case, the wobain lost the interaction with this bind site, ion bind site, and lost the affinity. So, for a good structure-active relationship, it's very important to consider the effect of potassium to characterize better the lactone ring. So, what we have looking for is a methodology to better characterize this interaction in the cheap way. So, remember that the sodium potassium TPase has both ATPase and phosphatase activity. When I working with all reagents necessary to perform the full cycle, we can see a phosphatase activity in the A2 conformation, mainly in the A2 conformation. So we have seen the literature that sodium potassium TPase can phosphatase a substrate known as paranitrophenol phosphate. So when this substrate is phosphatase by a protein at pH normally alkaline, this product, the paranitrophenol, has a yellowish color. However, in the case of our situation that we are working the pH 7.4, this color yet can be produced. So we can measure the activity of phosphatase activity of this protein in the potassium-dependent manner. So we just remove the ATP, remove the sodium, and allow the protein perform the phosphatase activity one with potassium, magnesium, and the substrate. So the main advantage of this method is that no need for additional protein, like some couplet system to quantify the activity, no need for additional reagent for quantification. The production of the reaction is chromogenic, and is allowed plate reading in a real time and a very low cost, which is very useful for our lab, because we have always working with a lot of number of cardiotonic steroids. So what we try to do in this situation was reproduce what Fedosova was shown with increase of potassium. So in this case, we have in the second column, we have the affinity of the cardiotonic steroids. I bring some examples of this. And we have the affinity in H2P conformation, that the bind site is more open. And in here we have the affinity and consequently the potential of these compounds in the phosphatase activity. And in a lighter section, we have the cardiololides, and more darker section, we have the bufadienolides. And what was seen in the when you make a ratio between these affinity these different effects of this conformation and a lot together with more of steroid cardiotonics. However, during the performance of this work, you have identified articles published by a group of Askari and Bagrov that they made questions about our information of equilibrium constant. So they say that the true affinity of the obaina is more reliable when they are in the close of the micromolar range. While we have shown that the obaina and together with a lot of cardenolides have a nanomolar range of the affinity. So in this situation, we have seen that there was a good situation to show the pharmacological points of view of this interaction. So what this author have did was pre-incubation of sodium potassium TPAs with 25 millimolar of potassium and then 100 millimolar of sodium. And after they start the incubation activity with 2 millimolar of ATP and 3 millimolar of magnesium. So what this author have shown that is when you have a graphical in the x-axis, y-axis, we have the sodium potassium TPase inhibition over the pre-incubation time. And what this author have suggested that the 10 minutes of pre-incubation is enough to bring the real equilibrium constant of this interaction. However, what we did was exactly the same graphical with the same situation, however, with only addition the time zeros in this way without pre-incubation. So what we are trying to say here cardiotonic steroid cannot bind only with potassium and sodium. They need at least ATP, magnesium, or inorganic phosphorus. We have seen this in the binding assay, and you cannot bind. So, but in this case, the actor has a functional time of the interaction between these two protein and with this protein and ligand. So 10 minutes is enough to bind in the equilibrium of sodium potassium TPase. What we have shown again was in the sodium potassium TPase inhibition over the different concentration of obain, that the time is very important for binding with the high affinity of the in this case of obain. So what we have shown here is that the time of incubation does not reach the equilibrium constant of obain. That is very important to better characterize every ligand that you are working with. We have published this situation with Professor Kalis and Adriana Katz. And however, it was interesting to take a look if this modification that you are working can affect the time of this interaction between cardiotonic steroids and sodium potassium TPase. So to take a look in the rates of the association, we use a simple interaction reversible model of this protein. So we assume that the interaction between sodium potassium TPase and consequently the inhibition follow a law of max action. When we have the interaction between these two components, we will guide it by a two rate of constant association rates and the dissociation rate. And in this case, we assume that the drug target formation is actually related to the inhibition. So in the equilibrium, we have the same values of the association and dissociation rates, and we can measure, in this case, of different inhibition over different concentration. We can see in the equilibrium, or we can measure the equilibrium by seeing the two rates of association and dissociation. So it's known that the interaction of the simple model is follow a one-phase exponential association equation. So what we have done and is well reported is adapted this equation to a interaction of drug receptor. So we have max inhibition, occupancy, description of the occupancy, and the rate constants observed will be described by the Booth rate association guided that is related to the concentration and the rate of dissociation in relation to the time. What we have is the inhibition of specific activity over the time, however, with the different concentration of this every compound that we want to measure. To bring this graphical, what we've done is identify the specific activity reduced from the basal activity, normalize the data by made a percent of the total activity. Consider that at time 0, the inhibition will be 0% to fix the basal condition. Fix the Imax to over to 100%. To not so we not expect over 100% of inhibition. And for fitting of this curve is necessary to share the both association and dissociation rate. So this interaction will bring the same association and dissociation rate by fitting all curves. So what we have seen is that the full cycle of sodium potassium TPase in the ATPase assay, we have the stress constant, two rates constant, and one equilibrium constant, and we have checked with the same situation with the partial activity by the phosphatase activity. What we have identified is that when you change the condition of this cycle, we have seen that the association rate of these molecules is reduced. So in the case of digitoxinine, this reduction of this association rate brings a reduction, an increase of equilibrium constant, as a consequence, reduce the affinity of this interaction. However, what we have seen for bufaline is that the buf association rate is reduced. However, the dissociation is reduced in a compensatory manner. So in this case, as we have reduced both dissociation and association rate, the equilibrium constant will not change. So it is the dynamically explain why the six-membered lactonic ring with the is made in the binding sites of this protein. So we have taken all these molecule, check this association and dissociation rates, and you perform a correlation between the equilibrium constant and the both association and dissociation rates constant. So we have a clear correlation for both situation, what is expected. However, what we are seeing is a situation, for example, for digitoxin and digitoxygenine. They share the same affinity, however, the completely different kinetic behavior. When you take a look at the results, we have seen that this both association and dissociation is completely different. And for example, if you want to see in this situation in the effect concentration curve, we are seeing in checking the different time, we are seeing that the curve of digitoxygenine did not change because the equilibrium of these molecules is very fast. on the other hand, when you take a look of digitoxin, when you take this curve and the different time, we see that this molecule take a long time to finally find the equilibrium of this constant. So in this case, again, it's the situation where you have the progression concentration over time curve. So what I see in this situation is the equilibrium of this AV concentration that we are using with the digitoxygenin is very fast to find. While in the digitoxygenin, the curve shown a very slow equilibrium situation. So to better understand this difference between these two molecules, we'll try now to test the energetic behavior of this interaction. So what we have done was try to understand the interaction of this protein ligand described by a graph of G's free energy over the binding reaction coordinate. So in this situation, we have, for example, the binding affinity described by the Gibbs free energy of two molecules that share the same equilibrium constant and consequently the Gibbs free energy. However, the difference between molecules that have a slow and fast equilibrium association is due to a large barrier between them. So a slower molecule will show a greater barrier, energetic barrier to perform the association. And this will slow the interaction between these components. So in this case, we have the description of the Gibbs free energy for the interaction of drug protein receptor, while the delta G describe the useful work energy available, in this case describe the spontaneity of the reaction. The delta H will describe the direction of the heat, is this component is endothermic or exothermic behavior, and finally the entropy that we describe the direction of this interaction. In this case, we can better describe the entropy as a microstate available for this interaction. So if not has the enthalpy, favorite enthalpy for this situation, entropy can be guided this interaction with the different temperature. So if you increase the temperature, the binding reaction can be occur. So in the case of the absence of this barrier can be described by the Eyring transition state theory. So in this case, we are talking about a theoretic structural interaction that we perform between molecules that can be dissociation of that will compete for the binding sites in the case of, for example, a desolvatation situation. And you can describe by the Eyring equation. In the case, the delta G is delta G, delta H, and entropy will be described always as a positive because we are talking about the energetic necessary to interact, this interaction occur. So what we have done is the same analysis, however, changing the temperature and made a linear mode of this Eyring equation. So what we have seen was the digitoxin, yes, have the same affinity at 37 points of degree. However, the digitoxin find this affinity only when you increase the temperature. What is not the true for the digitoxygenin. And when you check the thermodynamic components of these two molecules, we have seen that these have both the same delta G. However, the difference between these two molecules in the case of entropy. So digitoxin have more entropy than the digitoxygenin. Now we have checked the rate of association, dissociation of this interaction between these two molecules. You have seen that the association for both molecules increase with increase of temperature. So the only difference is the unity of this, the intensity of this increase. And when you take a look at this energetic compound, what you're seeing that is only the the entropy of the digitoxygenin is unfavorable. So in this case, we suggest that what the guide is association rate is probably the enthalpy, as the digitoxygenin has a disfavorable entropy, but binding much more faster. Now, when we take a look of dissociation rates of these two molecules, we have seen that when you increase temperature, digitoxygenin, they're more faster, dissociate with increase of temperature, but this is not the case of the digitoxygenin. That when you increase the temperature, the dissociation rate did not change. So in this case, again, this is explaining why this the different energetic behavior of these two molecules in the case of the association rate. So the digitoxygenin, in fact, is not affected by temperature because both association and dissociation increase. However, when you take a look at the linear mode of Ayring equation, we have seen opposite behavior. So when you increase in temperature, actually the dissociation rates of the digitoxygenin is becoming more slow. So we increase temperature and the dissociation probably reduce the dissociation. And in this case, in the Ayring theory, the enthalpy is always positive. However, in some cases, when it shows a negative enthalpy value, is normally suggests a complex mechanism of this interaction. So while the association rate is can be described by, for example, one model of association, the dissociation of digitoxin maybe can be doing the two-step mechanism. So this is what I have tried to explain. So we can see to try to explain this interaction between drug pharmacoreceptor, we can use some model to describe this interaction. So in this case, we have a simple interaction that can be described by only one association-dissociation rate. And we can, the induced fits mechanism, that very popular mechanism to describe the molecules that have a slower association-dissociation rate. However, it's very important to pay attention for some crystallographic results, because Kanai and co-workers have shown that the binding site of the sodium potassium TPase in absence and presence of many cardiotonic steroids did not change. So the interaction between these molecules do not induce, is not guided by induced fits. And probably this interaction can be described by the selectional conformity, as the affinity can be different by these two conformations. So as conclusion, we have seen that the potassium-dependent phosphatase activity through PNPP substrate show to be easy, cheap, and scalable way to perform a pseudoactivity and kinetic relationship. The 6-membered lactona ring can induce the compensatory reduction of the rate of the association, maintain the potential over the conformational change. Digitoxin and digitoxygenin have the same potential, however, completely different rates constant on potassium phosphatase activity. And digitoxygenin potential increases exponentially due to the reduction of the dissociation rate with the rise of temperature. So that's a complex mechanism for dissociation rate. So what we have doing the lab now is to back to some works that we have done about the functional selectivity of this interaction with these some compounds. In the case of telocinobufagine and marinobufagine, that we have seen recent paper that they have this different association-dissociation rates. So if you can describe the association, this kinetic behavior and the thermodynamic, can we correlate it in this time with the efficacy of this compound and describe the efficacy of the interaction between sodium potassium TPase and storage cardiotonic by kinetics and thermodynamics? So is the way that is the moment that we are trying to invest to try to describe this interaction. And of course, I want to thank our financial support, like a CNPq, CAPES, FAPESP, and recently FAPESP, and some institutes that have collaboration with us. So we are in the UFRJ, working with Professor Luiz and Professor François. We have working with Professor Leandro, Simone Silva, José Vilar. We have also worked with Professor George and Professor in the Israel, Professor Adriana Katz and Steve Kalish. And thank you. Thank you.
Thank you, Pedro, for your interest talk. Questions?
Thank you. Very, very nice. Now, I guess very difficult question, but all your modeling is based, you're working with purified enzyme.
Yes. Yes.
Supposedly that's the alpha beta dimer, right? 2 alpha, 2 betas. Maybe.
In the complex, yeah. We have working with the big kidney, so we have the alpha 1, beta 1.
So what do you think your models will change or had to be adjusted if it's 2 alphas and then you have 2 molecules binding? Do you think that there could be some allosteric effects, or and then that could modify the on or off constants, or?
In the case of the rate constant, what we are seeing some example of literature, we can change the association, dissociation, mainly the dissociation by changing beta subunit. So if you change beta subunit, in the case when you are trying to make different oxidic portion, if you increase the oxidic portion, we are now interaction with beta subunit, but not in the ground state, not in the, for example, the situation that you are seeing docking, because the effect of what you are seeing, the effect of beta subunit is the trajectory, is not in the binding ground state.
I see.
So when for example, obain and digitoxin try to dissociate of the binding side, the last oxidic portion interacts with the beta subunit. If this oxidic portion interacts time enough to restore the association of this interaction, so the dissociation will be very slow because the interaction between beta subunits. So try to dissociate, interact with beta subunit, and become association again without fully dissociation.
Yeah. Very nice. Yeah, thank you.
It's the main example. Is allosteric effect I can now.
After, so maybe you can continue now, and then after that you can speak with him. Yes.
In the case with the American, we need to separate.
Maybe so go ahead. Yes. All agree?
To check if there has a allosteric effect.
Yeah. So now, thank you. Thank you, Pedro. So now I want to introduce Dr. Giorgio Doherty. He's a full professor of chemistry at the Department of Chemistry in Northeastern University in USA. And his research group has been working to related areas in organic synthesis, the carbohydrate synthesis in natural products. And today he's going to talk about carbohydrates and cardiotonic steroids. Thank you.
All right, great, thank you. This is a great pleasure for me. I always have a fantastic time when I come down to Brazil to see old friends, new friends, and to tell you guys a little bit about my science. I am keenly aware that there's probably a minority of synthetic organic chemists in the room. And so normally I'm nervous because you're going to worry about the stereoselectivity on step 7. Now I'm more worried about the fact that a lot of this stuff just isn't that interesting to you. Though as I watch this audience, the fact that you can see this complex biology and then pay attention to beautiful kinetics, it's really impressive to the depth of chemical science this community enjoys. And so I'm going to be quick. I'm not going to worry about lots of details. I'm going to try to give you my perspective as a synthetic organic chemist and how I sort of apply that to medicinal chemistry. To be honest with you, almost all of our projects really get started with just a want, a need to make a molecule. Maybe we think this new reaction might work. Maybe we think this gives us novel access to a molecule. But we're a research group that depends on funding, and so we always try to find a biomedical applications. And the cardioglycosides are probably our best project. I'm going to introduce sort of our approach to carbohydrates, because that's really the type of medicinal chemistry we focus on, is carbohydrate medicinal chemistry. I'm going to show you some non-cardioglycoside sort of examples. Then I'm going to show you sort of the structural depth that we can get to in terms of carbohydrates. And then I'll show you the kind of cardioglycosides we make. Okay, and so in terms of what we think about, we really think about asymmetry, and we really think about natural products and the role of natural products play in nature, medicine. Okay, and then just because I know we've been here for a while, I'll give you the results now. And so if you want to doze off, you can do that too. So this really represents and sort of our best results in terms of cardioglycosides. Over the years now, we have essentially been trying to reverse the effect of the Chapos brothers, who bring organic chemicals from South America to North America. We have been trying to reverse that process, and we've basically sent a lot of these compounds down south to Luis's lab and Leandro's lab, and we'd like to get more molecules to you. So please feel free to contact me. And so essentially what we've been able to do is take a molecule you guys probably know more about than I do, digitoxin, and we've been able to change the sugars. And today, what I think is our most impressive result is we've been actually now finally been able to find an oligosaccharide that has improved activity in terms of, and I should be clear, when we're talking about activity, we're pretty much using a cancer cell toxicity assay, and then we have some CMV viral antiviral assays. And so we can actually improve the activity of the cardioglycosides by, if you think about it just from a structural point of view, all we have done is add 3 oxygens to this molecule to get to this molecule. But to do that, then we basically break all the bonds and put them back together with a different stereochemistry. And I bet everybody in the room has taken an organic chemistry course, and you remember playing with the models. I still have great satisfaction to hear the popping and clicking of bringing these molecules together. And you can do that with the models, but the reality is you need complex organic synthesis to do this transformation. In terms of the monosaccharides, we've been able to show that if you go from a beta D sugar to an alpha L sugar, you can improve activity. And you can particularly improve activity when you have a rhamnose or an imisotose. So 2 X's here are either OH or hydrogen. And then if you put an amine at this position, you can really, that's probably our most active compound. Okay. And so, a lot of hullabaloo has been going on in the United States for the last 8 years. And as that has happened, we have benefited from some things. And one of the better awareness of some of our societal problems. And so we as a community are probably just more aware of our sort of inherent biases, okay? And the inherent bias that I'm most keenly aware of is chirality. We live in a 3-dimensional world, and the reality is if you start to explore the 3 dimensions, you constantly create chiral situations. So you can think of yourself as symmetrical. If you're a cartoon character, you're symmetrical. But if you're really alive, an artist will tell you, if you want to make a scene look more real, you'll break the symmetries. Okay? And so nature does that too. Why? Because it just needs to take 3 dimensions. And if you make chiral molecules, you have more opportunity to make molecules. And if you only make 1 enantiomer of those chiral molecules, you just have a simpler job once you've solved the big problem of picking which enantiomer to go after. But nature, long before we were around, has made this choice. And so DNA is a very complex structure that's chiral, and its building block is chiral, and the chirality comes from its building block. Proteins are chiral similarly, and their building blocks are chiral. And so proteins recognize proteins, DNA recognize DNA, but they really have sort of limited sort of interactions if you think about the 3-dimensional space. And natural products actually play this really cool role in the sense that, at least my vision of why we put or why basically the simple organisms, bacteria, do so much secondary metabolite synthesis. It's really to fill the gaps that are within proteins and in some cases DNA. And so it just explores new space. So you have a chiral protein, and you think about how a beta sheet interacts with a beta sheet. That's very specific. But if you think about the spaces above and below or between 2 alpha helixes, there's really lots of unexplored space that an amino acid maybe not be the best building block to fill that space. And so natural products have the advantage that they come from achiral starting materials. And so you can get to more different structures. The problem with this approach that nature finds is the reagent that it uses to do this are enzymes. And enzymes are proteins, and proteins are chiral. And so when an enzyme has figured out how to do a reaction, it's always chirally based. And so the reality is if you're going to have a steroid, you're going to get this enantiomer, not the unnatural enantiomer. That's true with the polyketides. And then when you think about the carbohydrate versions of these molecules, it gets even harder because nature actually makes all of its sugars from glucose, whether it's D or L. And so it's a multi-step process, and at each step there's a chiral enzyme. So when nature figures out a way to go from a D sugar to an L sugar, it really can't make all the possibilities. Okay? So this is really a limitation of nature. And if you think about it from a medicinal chemistry point of view, an excellent opportunity for synthetic chemists. We believe we can get to structures that look like natural products, but they're structures that nature probably never got access to. And if I believe in the awesome power of evolution. I don't live in Kansas. But that awesome power of evolution does not get to apply if it didn't get to ask that question, right? So if you don't have access to that structure, you don't know if that structure is a beneficial or non-beneficial change. Okay, and just to remind you, the chirality is everywhere, right? So we always learn butane is an achiral molecule, but it only is achiral in 2 of its conformations. When it's planar at these little points, it's achiral. But as it turns, it's actually a chiral structure. And so actually, when we think about any structure in life, it's really chiral. And then when it's in an environment of a protein or DNA, it's really biased in one way or the other. Okay, and so we always learn about carbohydrates, we get Fisher projections, and we learn about chirality, and then we start going from the simplest sugars to the hexoses. This is, don't worry about the details, but the thing to recognize is of all the possible structures, really nature only uses 1 of the 5 carbon sugars and 3 of the 6 carbon sugars. And so when you cyclize these, I should just tell you, just to remind you, glucose, mannose, and galactose. And when you cyclize the structures, you create greater complexity, but you really only do that from the simple starting material. So there's really just unused space, unexplored space. Now, if there's any bacteria in the room, they will tell you, listen, George, we've been doing this for years. So natural product biosynthesis that occurs in bacteria, they are much more adventuresome in terms of sugar substitution and stereochemistry, but they still are limited by the fact that they have chiral reagents. And they basically will never make the enantiomeric chiral reagent. And a synthetic chemist can. Okay, and so then this is how we think about making molecules. Right, here's a natural product, D-swantonine. It's a famous natural product that was discovered from cactus. Cows that were eating the cactus would go mad. Basically, they had a natural product in there that inhibited the reprocessing of the carbohydrates on proteins, and that causes essentially death eventually. And so we worked out an asymmetric synthesis for there, starting from this achiral starting material. The advantage of using asymmetric catalysis means that we could make the enantiomeric material. And in the case of swantonine, actually the L-swantonine then becomes actually a rhamnose inhibitor. And so that's a potentially useful thing. In fact, if you buy L-rhamnose from Aldridge, it probably came from our lab. Okay, there's other examples of this. And so we're always looking for how we can explore stereochemistry and medicinal chemistry. And to be honest, nature does do a lot of this stuff ahead of time. And so more often than not, we're going to find that the chiral structure is the ideal structure. And so here's a good example. Here's a molecule that's discovered from avocados. It exists in avocados, but it really is highly prevalent in the seeds and skins of avocados, and it has some interesting anti-cancer activity. We got interested in it because it's a molecule where the triple bond is important for activity, which is crazy when you start thinking about it. And so we wanted to explore that, but we also wanted to explore the stereochemistry. And then so from an asymmetric synthesis point of view, if you could think about trying to make this molecule from an achiral starting material, then use a chiral reagent to induce one chiral center, and then with that chiral center, control the next chiral center, either cis or trans, and then you could make the natural products essentially with a double bond, with a triple bond, or saturated. All you have to do is use the enantiomeric catalyst, and you can get the enantiomeric. Right? Nature, there's lots of examples of nature's having enzymes that can, let's say, hydrolyze an ester on an alcohol, and you can get the one enantiomer versus the other enantiomer. But when you do those sort of evolutionary studies, you never get the enantiomeric protein. You get a different structure, but it's going to be containing L amino acids. Okay. And so we sent the compound north of the border to Canada, and they screen them, and what they find is actually you need the right stereochemistry to have the right activity. Okay, so here's another natural product that got interesting to us. It's another polyketide. We got interested because we thought we could make the molecule from an achiral starting material where we would induce one chiral center here, then do a diastereoselective epoxidation. And then a friend of mine from Stanford developed what we call the Coates carbonylation reaction. And we thought that would be a really cool way to approach this molecule, especially because it's a friend's reaction. And so he developed this catalyst, which we call the Coates catalyst. And so we could not make all the possible structures, but we could make a good series of steroids. And particularly, it's really not surprising that this four-membered ring, you're familiar with beta-lactam, well, a beta-lactone is obviously going to be a reactive species, so you assume that's a pharmacophore. And so we really wanted to explore the stereochemistry around that. And so we make a bunch of molecules. And then we test them, okay? And so tetrahydrolipostatin is actually used in the drug. I think it's in the US, it's called like Ally. It basically inhibits a pancreatic lipase in your gut. And so essentially, though you want to eat a fatty diet, you de facto have a less fatty diet if you ignore the problems in the bathroom. And so it wasn't surprising to us that this might be a case where the stereochemistry is matched, because the reality is that the organism that made the lipostatins was never thinking about human pancreatic lipases. Okay, it's thinking about inhibiting a bacterial lipase to get some sort of advantage as it's growing in its environment. And so we screened it. Now, it turns out this is actually the best lipase, but it's surprising how good the other structures are in terms of their activity. And really surprising to us is that you take the pharmacophore and you make the enantiomeric or local enantiomeric structure, and you still have some pretty good activity. So if you change two stereocenters, you can actually get some pretty good activity. And so what that really says is this is the example where nature hasn't fully screened it, because nature has never even screened the molecule. Okay, and then when you actually look at the same set at the bacterial targets, and then you don't see that effect. Now the enantiomeric lactones are not active at all, and so you go from nanomolar to micromolar. So that makes sense. And these are lipases associated with some sort of microbacteria. Okay, so that's just natural products in general. Now we're talking about carbohydrates. we really became carbohydrate chemists because we got very interested in trying to make chiral molecules with lots of stereocenters efficiently. And we knew about the Akhmatovich rearrangement, which is the rearrangement of a furan alcohol into a pyranone, and that pyranone, when R equals H, is a sugar called acylose, and we could think of turning this enone into other polyols, and so you can imagine making glucose, mannose, galactose, etc. -ose. And then the key feature here was you could make this furan alcohol then from an asymmetric reduction of this ketone, and again, that same catalyst that was before, let's not worry about the details, you can get to either enantiomer. In this particular case, it is really highly enantioselective and very scalable, really practical. We can make hundreds of grams of these sort of materials. So then you do the rearrangement, you then have to do an acylation, and so this chiral center can control this one. In our case, we actually don't try to control it, we want both isomers. But from a carbohydrate point of view, and when we learn about sugars, right, there's 4 stereocenters in a sugar, so, and then when you make the lactone, there's a fifth, 2 to the fifth, so there should be 32 possible structures. But when you simplify the structure to 2 stereocenters, then there's really only 4 possible compounds. So cis and trans, and then both D and F. And then the key feature here is we have, I should go back, this is a very stable glycosyl donor. So then we can make a lot of it, store it, and then go forward. Okay, there's steps. And so the eighth method, we have to do steps, and we do those steps. Oops, how did I do that? Sorry. Okay, and so then we can take that anomeric stereocenter, and what we can do is use palladium 0 to ionize this allylic position and trap it with the nucleophile. And essentially, the palladium inverts the anomeric position with inversion, like an SN2 type process, and then the nucleophile comes in an SN2-like process. So double inversion means retention, and so our alpha starting materials give us alpha products, and beta gives us beta. So that then implies that we can use this quite generally. And then, this is not so important to you guys, but a key feature for our discovery was that this ketone really gives you a really hot electrophilic glycosyl donor. So it really works with a range of alcohol nucleophiles, which has some real advantages, and it works in all the possible stereochemistries. Okay, and most importantly, at least in the alpha, or we'll say the trans series, you can always reduce this ketone and dihydroxylate that double bond and get to mannose, rhamnose. And it's really, really scalable, really practical chemistry that works with fantastic diastereo control. Okay, and so stereo control in this concept basically makes the synthetic transformations easier because you spend less time doing column chromatography. Okay. And so from a carbohydrate perspective, it's kind of new concepts here. Essentially, you have this palladium pi-allyl as being like an anomeric directing group. And then a key for us is the fact that that same enone kind of serves as an atomless protecting group. And so normally in sort of synthesis world, we think of taking functional groups and putting on blocking groups and then doing a transformation, taking off the blocking group. And that steps takes time, you lose material, and then you generate more waste. So there's some potential environmental advantages to this sort of approach. Okay, so here's some examples of like monosaccharide medicinal chemistry. Here is an antibiotic called pyrimidine, which is a really interesting antibiotic from my point of view because it inhibits the ribosome. And the ribosome, of course, is essentially, if you think about it, it's really just RNA. So it's a polyphosphate, very anionic material. And it is really rare to think of a polyketide without any charges as being a good ligand for that ribosome, but it is. And that's the cool thing about nature. You would never think of developing an inhibitor of an enzyme that binds sodiums and potassiums with a neutral polyterpene, but it does, okay? And so what medicinal chemists have found is that if you put these six-member rings with amines on there, you can improve the activity, which makes sense. Ribosome is negatively charged phosphates, and amine is protonated or positively charged, so plus likes minus. Not the hardest thing in the world to figure out. And then we got interested in the idea that you could essentially mimic a lot of that electrophilicity with these enones. And we did some modeling, and we convinced ourselves that these structures might overlap where that structure is, and so we wanted to test that out. So because we could also make the molecules, we did the design this way too. Okay. And so this was the basic idea, and then the stereochemistry matter. Okay, and so essentially we develop what we think of as a new unnatural class of natural products. We call them saccharomutilins. And we make them and test them. So in terms of the pyranones, we can do all the possible structures. And what we find is, yes, we can actually get some pretty similar activity to the lefmulin natural product. And actually the other one, the bicyclic one, it's actually almost identical in terms of its activity. And then most importantly, stereochemistry matters. For us, when we don't necessarily care what stereochemical is the most active compound, but if stereochemistry matters, then we know we're sort of in a active site near an amino acid or in this case, RNA, because those sites are chirally biased, okay? And so that kind of tells us something. Okay, and so we can also reduce the ketones, and we basically are looking at all the possible saturations because we can. These molecules are actually pretty good, so they actually have some pretty good anti-cancer activity, antibacterial activity against, and we also you can essentially isolate the ribosomes and do it in an achiral, so you can show that the ribosome is the target, and it is. And it doesn't affect eukaryote ribosomes, which is pretty important in terms of drug development. And then medicinal chemists hate enones. They call them pains compounds. And so, and they were right. There actually is some cytotoxicity associated with these structures in human cells that would presumably not be ribosome related. But interestingly, we can reduce the ketone and get rid of that concern and keeps a lot of that activity. So that's pretty exciting for us. Okay, so we think we can put monosaccharides on natural products and do medicinal chemistry. Okay, this is not a new discovery. Men have been doing this for hundreds of years, or 100 years, and nature's been doing it for more years than we understand. And but what nature does that men don't do well is really do that same kind of structural study at the oligosaccharide level. And the reason why is it's hard to make the monosaccharides, and then when you start stitching them together, it just gets harder. Steps are bad. Okay, but we thought we kind of had a unique way to go at oligosaccharides from these same achiral starting materials, and we thought we could do it in a way that would be some step efficiencies. Okay, I thought I pushed the button. Yes, I did. Okay, so I'm skipping steps, but essentially that same palladium glycosylation reaction you can use to make this bond, and this bond, and that late stage reduction in dihydroxylation you can do all in one pot. Okay, so that's the step efficiency. The fact that you can do it really means that these reactions are highly selective, because if they weren't selective, you would have diastereomers, and that would be a chromatography nightmare. Okay, and then you can go in the 6 direction, you go in the 4 direction, so you do the reductions after. You do the reductions after the glycosylation and the dihydroxylation's late, and they work really well. So that atomless deprotection is working locally. It doesn't worry about global stereochemistry, which allows you then to think about these things in a better way. So you can go 6 and 4, you can also do them at the same time. So you can make pretty complicated branch structures, and then you can do that dihydroxylation. And you can make that. This is not, I mean, a carbohydrate chemist would tell you that this is not that hard of a structure to make. They're showing off when they say that. Okay, and so we also think it's not that hard of a structure to make. But what they would tell you is that this is almost an impossibly hard structure to make, because these double bonds make these anomeric positions very acid sensitive, and the traditional glycosylation reaction is very acidic, and so it just wouldn't survive. The way you would make this molecule is actually removing these oxygens to get to this sort of structure. Corey Winter olefin synthesis, if you're into it. Okay, and then the enantiomeric structures would be much harder to get at. Okay, and then we can also go to other structures, right? So the late stage divergency is really what kind of inspires medicinal chemists to think about these structures as interesting. Okay, and then convergency, so we can transfer this group late and then branch. Okay, so we can do 6 and 4, but you really can't branch in the 3 and the 2 direction with these double bonds, because you need alcohols there, and the double bond isn't an alcohol. And so we had to figure out a way to go after other structures. And so the 3 direction for us involved protections, but the protections just didn't work out for us for this particular synthesis. And what we basically figured out was a way to use this boron catalyst to really selectively put the group in at the 3 position. Okay. And so then, and you can put it on either stereoisomer. And depending on what's at the 4 position, you can get either good control of the 3 position. And in one stereochemical series, you can get 2 positions. The other one is not that good, but we're working on it. Okay? And so don't worry about these details. Essentially, though, in terms of the 4 possible regioisomers, we can do 3 really well, and this 4th one has a problem. Okay? But if you can get them on, you can then do the same deep protection, if you will, reduce and dihydroxylate, and that works really well. Okay, and so we'd like to boast a little bit and suggest to you that this plating glycosylation reaction really has all the hallmarks of a glycosyltransferase. And if you're into sort of catalytic efficiencies, the glycosyltransferases are the bee's knees. To be able to make a glycosidic bond on a polyol specifically, regiospecifically at one position and stereospecifically, and to do that in water is just amazing. Okay, but we can mimic a lot of that in the sense that we can get regioselective glycosylation. And what we can do that nature can't do is we can take 1/2 and make it the enantiomeric half, because our catalysts are achiral. They're not biased by that. The only chiral aspect of this interaction is the 2 chiral subunits coming together. But in theory, nature would have that problem too if it was evolving these structures. Okay, and then essentially what that is telling us is that depending on our substitution, we can essentially ring flip this to get opposite selectivity. But let's not worry about that. Okay, and so we can also, instead of transferring the pyranones, we can also transfer these azido sugars, which will be important in the end because that's going to be a C4 amine that I told you was important. Okay, and so don't worry about that. Okay, so why I'm here to talk to you today. We can do medicinal chemistry on the cardioglycosides. Why? Well, earlier this week, we mentioned the name Jack Kaplan, and Jack Kaplan would disagree with this, but there's some pretty good evidence out there that there's some really potent anti-cancer and antiviral effects associated with cardioglycoside. I believe Kaplan has a paper that basically says, yeah, but they're more toxic to non-cancer cells. But that is true for all cancer drugs. And the reason why is these cell line assays are such that if you don't have an immortalized cell, it's just harder. They're just more sensitive to everything, right? So they're more sensitive to sodium chloride also, right? And so I think there's good evidence out there that there's a window of activity associated with these molecules. And in terms of how we screen them, because we have a limited ability to do biology, we can grow cancer cell lines and we can do MTT assays. We can do that pretty reproducibly. And so that's where we do all our screening. And then with collaborators, we do other things. Okay, and so this is sort of the signalosome hypothesis. I won't explain it to this crowd because you guys are the ones that explained it to me. We can do some of the world's crudest modeling. Our problem with modeling these structures is any kind of minimization you do, it literally ejects the cardioglycoside. So we don't do minimizations, but we do space filling kind of ideas. And we've convinced ourselves that that's changing D to L sugars makes sense. Okay, but really, we're natural product chemists. We want to make the molecules that nature makes. So we wanted to make digitoxin, and to do that, we were going to take digitoxin in and put on the sugars. The reality is, from a price point of view, digitoxin is much more expensive than digitoxin. So what you really do is you knock the sugars off and you put them back on. Okay, so it's not the most efficient synthesis. It's negatively efficient. Okay, but it's stereoselective. We can very stereoselectively put in to get you the digitoxin stereochemistry. You can do that without any protecting groups. Okay, and then you can essentially put 1 sugar on. You just repeat. You have to do a selective acylation. You repeat the process. You put 2 sugars on, and you can repeat the process and get 3 sugars on. Actually, the more interesting structures are the monosaccharide and the disaccharide because you can't selectively hydrolyze digitoxin down to the mono and di. So those are really synthetic targets where it turns out we just buy digitoxin. Okay, and so put on 1 sugar takes 5 steps, 2 sugars takes 10 steps, 3 sugars takes 15 steps. And so if you're going to make a library of structures, you're focusing on monosaccharides because doing a lot of 5 steps is easier than doing a lot of 10 steps. Okay, and then medicinal chemists love these sort of things. You want to just do systematic structural variation. We call this well, I won't bother with details, but we can essentially take the C6 position of our most active structure and then explore it by making the methyl group an ethyl and propyl. We can then branch and fill up space. And you quickly realize that there's kind of a tight interaction here. You can go from methyl to ethyl, but anything larger loses activity. Okay, and then again, we can essentially explore that monosaccharide by essentially popping off the oxygens and moving them around and inverting the stereochemistry. Trivially easy to do with a molecular model kit. The reality is to do these sort of inversions, really each one of these molecules is a unique synthesis. Okay. And then we explore those for activity. And in that process, we basically have convinced ourselves the most active compounds are either rhamnose, R equals methyl, or these two groups are hydrogen, and that's imidazole. And those are, depending on the assay, I would say 5 to 10 times more active in an anti-cancer assay than digitoxin. Okay, and just to show you that the data is real, this is the kind of stuff we love to do. You put a D sugar on, you put a D sugar on or an L sugar on, and you get different activity. So it's not surprising, the sodium potassium pump is a very chiral environment, and so the sugar stereochemistry matters. Okay, importantly, so this is synthetically how we do that. So we put on the sugar, we reduce, and we either dihydroxylate or we do this diamide reduction. And so essentially from one compound with one given R group, we can get 2 different structures. So that's nice. Oops, that's a typo. That slide shouldn't be there. We can also take the imidazole or the rhamnose structure, and again, using steps, we can put in an azide, and that allows us to get to these amino sugars. And these are by far our most active compounds. C4 amine, and our modeling kind of suggested this, that there was an important hydrogen bond to the C4OH group. C4 amines are another, let's say, 5-fold more active. Those are rough folds, if you will. But these are the most potent, I think, digitoxin cardioglycosides. The reality is making these things more toxic isn't that important, but you have an assay, you keep driving in that direction, type of a thing. Okay, turning off activity and selectively turning off activity is what we really want to do. Okay, and so one way to do that is actually with prodrugs. So in the digitoxygenin series, we can essentially glycosylate and then acylate the C-ring alcohol and install rhamnose or imidazole. And depending on what that group is, you can essentially turn off activity. Okay, so these are sort of the range of cardioglycosides we've made, and then when you start putting on bigger R groups at the C ring, you can essentially turn off the activity. These are not in any way sodium potassium inhibitors. The trick now is so we can put a pivolate on, but then you can't take the pivolate off. You can put on like nitrophenol carbonates, and those can be photochemically cleaved and things like that. But what we're really looking for is some sort of environment that would be unique to where sodium potassium pump inhibition would give you a selective response. And if we could then think of a chemical reaction that would make a molecule, if we could make a molecule that's sensitive to that chemical change, you can imagine selectively targeting sodium potassium pumps. So we basically have the technology. Now we have to just think about the application. Okay. And so the other form of selectivity you guys know about, and that's isoform selectivity. Okay. And so to be honest with you, we haven't really exhaustively explored all the monosaccharides, but we have never found a monosaccharide that had any improved isoform selectivity. Okay, you improve the activity, but it's pan-isoform. And so we convinced ourselves that that's not going to be true with oligosaccharides. Okay, again, you start a project, you're going to make the molecules, you have to convince yourself. Okay, but nature's already shown us that. Actually, if you look, and this is Karolish's data, if you look at digitoxin, monodi, and trisaccharide, what you'll see is the isoform, you make the trisaccharide not to be more active, it's to be more selective. Okay, and so, and he has tested some of our 1,4 oligosaccharides and has suggested that there is some selectivity there, but we're really drastically turning off the activity, and there's some isoform selectivity, so it's not really the perfect setting, but hopefully we'll have gotten back to that question by the end of the talk. Okay, so I showed you how we can make 1,4 oligomers, and so we did that. And so the cool thing is we can make them all D, we can make them DLD, et cetera, and then we can also diverge by a dihydroxylation or reduction. And so we make a whole bunch of, we're pretty exciting for us to make an oligosaccharide library of structures. Even though it's 12 compounds, it's still pretty unique. Unfortunately, anything you do to these monosaccharides, you essentially turn off activity. And we kind of knew that pretty early on, but for completeness sake, we showed it was true for all of them. And we knew that because that OH bonding in the 4 position was pretty important. So putting a group on the 4 position, in retrospect, wasn't the best designed approach. So you go back to that same model and say, well, if you can't do the 4, how about the 3 or the 2 position? Well, we recently developed chemistry to put sugars on the 3 position, so we did that to explore the 3 position. So using this boron chemistry I told you about, you can put the pyranone selectively at the 3 position, you can put D or L on, and you can reduce dihydroxylate or reduce again. So you get 4 new disaccharides and you test them. In our anticancer assay, we go from 35 nanomolar to one that's, I'll call it the same activity, right? So we basically can take this molecule that's very chirally sensitive, put on a second sugar, and still show that the structure of the sugar matters, okay? So we're still in the environment that we were looking at. You just repeat the process, right? And so you just put on another D in your L sugar, and you test them, and you find that the L one works better than the D, and so you have a trisaccharide, okay? And essentially, remember, we went from digitoxin trisaccharide to a D sugar trisaccharide to an L sugar monosaccharide, and we improved activity. And now we've been able to build the trisaccharide without losing activity. Importantly, it matters in terms of its sort of structural design. So you go on, you do this again to the 4th sugar, and essentially now you're just losing activity. So it tells you, at some point you go too far off. You're either bumping into something in terms of this loop of the external loop of the sodium potassium pump, or you're just out in water, and that's just the effect of hydration. Okay, so we kind of realized we should stop there in terms of growth, but we're not done yet, right? So this is really just the starting point, right? So now we want to derivatize this. Well, we want to really test these in an isoform selective assay, but then once we do that, we want to start playing around with medicinal chemistry here with the idea that can we find substitution that would selectively turn off activity? Okay, if you could do that, I think you have something. Okay, so that's sort of where we're off into the future. And so that's sort of our medicinal chemistry approach, but it's carbohydrate medicinal chemistry brought to you with no added sugar. Okay, and if you watch TV in America, that's very important. Okay, so I think I'll stop there. I should really thank these people for doing all the work. I should thank my collaborators, funding, and most importantly, thank you guys for the opportunity to come back to Brazil. Thanks for staying awake. I really appreciate that. And thanks for this lovely week. I'll be happy to answer any questions.
Thank you, George.
That was fast.
Questions?
I have a very simple, thank you very much, it was exciting, but I have a very practical question. Will George, the creator, get my wish list?
I don't know.
For future compounds?
Yeah, sure. I mean, absolutely. I can tell you essentially the...
But I have one specific,
okay, but
which I'll tell about this in my...
joke to tell, if you give me 1 second.
Mm-hmm.
So we have this illegal drug pipeline coming down too. So I have Brazilian connections. And so whenever a Brazilian goes from Boston to, I assume, Sao Paulo, we usually try to get 1 sample into their luggage. And so I think right now, like all of our compounds are in Brazil, but the ones we talked about, we definitely can make more. We'll gladly send them to collaborators. And then we're really keenly interested in the idea of if you have a design question that we could potentially address,
yes,
we would love to work with you.
Yes, thank you.
Hi, Professor. It was a nice, very
He's a
talk.
user.
But I cannot work without these molecules. So, Professor...
Sign of addiction, in case you're wondering.
Yeah. Professor, I think what I was asking is a little distance what you have done, but in the case of steroid core of these molecules, you can made it by de novo as you done with this osteolytic portion, because I think it is interest to check what is the limit of this steroid core to start to add new sugar, but by removing some parts of the storage core.
I mean, so synthetically, this is a much harder target. Though, I mean, nature would tell you this specific one is easy to do, but once you start varying, I think it's a great idea. We definitely should explore that. And particularly, you could imagine, if you could imagine a structure where you've put a sugar here that you've significantly improved activity, you could imagine simplified structures.
Because in the case of this interaction between these molecules and the protein, if you always increase the sugar portion by the number, we are always increase the conformational flexibility of these molecules and always increase the entropy of these molecules. So if you now you go into reduce the size of these molecules, so you can try with the focus of this storage core, remove for example the lactona ring and work with this modification of this storage core by de novo. Is possible or is distance of this?
So John would tell you you can do a bit here. Like they've actually converted this to a furan. Those are pretty interesting compounds. We'd like to explore those with our sugars. John will explain that. Right, but so there's two aspects that make me think about this from John's perspective. One, there are definitely things we can do with the furan. What we'd really like to do is think about simplifying this structure. I say that because I came to this perspective thinking that essentially nature designed the steroid core to specifically inhibit a sodium-potassium pump. But if you believe in endogenous wellbeing, and I'm encouraging you to think along this ways, you could actually think it differently, that actually nature evolved the steroid structure and the sodium-potassium channel pump at the same time. And if that's the case, then it'll be a little harder it'll be more selective for the specific structure. But the only way to really explore that, I think, is by structural variation. So I'm 100% with you. It's difficult chemistry, so we have to think about it. My thinking would be to try to keep these two rings together and keeping sugars together and then putting those two ends.
Okay. Thank you, George.
Excellent. Thank you.
So now you had the coffee break, please. 15 minutes. 15 minutes. We will try to start at 3:20. Thank you.
Yes, 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 it's
So now, let's continue. I have also the pleasure to introduce Dr. Natalia Fedosova. She is Associate Professor of the Department of Biomedicine in Aarhus University in Denmark. And the main research interests of Dr. Fedosova are within the structural functional relationship of P-type ATPases, primarily the sodium potassium TPase, and also the interaction of cardiotonic steroids. So thank you very much for your presence.
Okay. First of all, I would like to thank organizing committee. It's spread all over the place, organizing committee, and you, Luis, for a nice presentation, for very kind presentation of me, and audience, all of you, because you created such a constructive, productive, and very warm atmosphere during the whole meeting, I think. I enjoyed it. The title of my talk, as you can see, the first sentence is with a question mark. It somehow reflects the ambiguity which always surround application of the cardiotonic steroids in the clinics. And then I would like to talk to you, how do we approach and try to solve this issue? And as it's a custom at this conference, I would start with a little bit of history and start with saying that cardiotonic steroids, cardinalides, were first introduced into Western medicine by this guy, William Withering, when he in 1785 published his paper, an account of the foxglove and some of its basic medical uses. And this event was surrounded by a bitter and protracted dispute with the grandfather of Charles Darwin, Erasmus Darwin. Both physicians claimed to be the first to apply infusion of the digitalis purpureae, early foxglove, to their patients. And at the end, Withering won. And just to state his victory, he ordered this portrait where he sits with a foxglove in his hand, and the book right next to him is open on the page describing this plant. So this plant contains, as we know now, mostly this compound called digoxin. So this plant was accepted as a medicinal herb long before people realized that it has an inotropic effect on the heart, and long before the target protein for this for this compound was discovered. It was initially used as diuretic, and only later physicians found out the connection between excessive accumulation of a fluid and a weakened heart. Later, it was the inotropic effect was ascribed to a change in the potassium transport, and as Gustavo told us in 1953, Schatzman showed that ouabain, which is a relative of the substance, found out that it inhibits potassium uptake and sodium extrusion in erythrocytes. And 4 years later, Skow discovered sodium potassium ATPase, and since that time, the sensitivity to ouabain, cardiotonic steroids, is a hallmark of the sodium potassium ATPase activity. This slide, I think it's redundant now, but I'll just keep it for a flow. Sodium potassium ATPase is very important, and it actually consumes about 25% of ATP reserve in a normal cell. In kidney, it comes up to 40%, and actually in brain, all related sodium pumping activity can reach 80% of ATP reserve. So all this energy is used to maintain gradient for sodium and potassium, and by this way maintain resting membrane potential, excitability of the cell, cell volume, as well as supply energy source for transport of nutrients. Here I show amino acids and sugars, as well as transport of cations. And here we can see that that actually counter transport of sodium and calcium is an basis for the inotropic effect in the heart. So this is here we are talking about cardiotonic steroids from cardenolides family, which means they all have 5-membered lacton ring, and they were first introduced into the on the western part of the world. In the meantime, in Eastern Asian region, in their cultural sphere, the physicians used extract from the toad skin, which they call chansu, and this is Chinese characters Hansi for the chansu, which is enriched by this substance bufalin, and as you can see, they have almost the same characters in their as when they're written. So this is bufadienolide, as we can find out from the name. So it has a 6-membered lacton ring. Egyptians, ancient Egyptians, they used this plant. It's from a Scilla family. They used powder of this plant also for medical purposes. And it is enriched by this compound also, as you can see, bufadienolides. So bufadienolides originally were for 1,000 years or so, or maybe more, used as a remedies against cell growth. And this is quite understandable because, as we know, sodium potassium ATP is a critical enzyme, and they are inhibitors of the critical enzyme. Everybody is aware of very narrow therapeutic index for decoxin, so it's clear. Until 67, the report appeared where authors claimed that certain cardenolides had a very, very strong cytotoxic effect on the cell lines, but had no effect whatsoever on the Ehrlich-Ascites tumor, not at all. That puts a question: what are the mechanisms? What could other mechanism could be? And it started a long range of different experiments. People studied a lot of different cell lines, found out a lot of different ways of explanation this cytotoxic effect, and it led to a fact that in 2005, the China Food and Drug Administration approved chansu, so extract of this skin, toad skin. Chansu is in encapsulated form for application in combination with conventional chemotherapy. They say that in some cases, in some cancer types, chansu improves action of conventional chemotherapy, and at the same time decrease the negative effect of this treatment. This slide you have seen many times. So we have cardinolides, 5-membered ring, bufadienolides, 6-membered, lacton ring. And I just mention some things because I'm going to use them in my talk. This is steroid core. So this cis configuration leads to a existence of the alpha surface, which is hydrophobic, and beta surface, where we find all the substituents, and they are more hydrophilic. What is interesting for us, this is position carbon 14, where we have this hydroxyl group, and this is important for interactions with the sodium potassium ATPase. And I would like to talk about position 3, where we find either hydroxyl group or very often glycosylation sugar residues, which they are not mandatory, but they have a very big large importance for interactions with the sodium potassium ATPase. This slide you have also seen many times, and we learned a lot about it. So how to explain multiple effects of the cardiotonic steroids in the cell, assuming that cardiotonic steroids, all of them are specific ligands for the sodium potassium ATPase. We can suggest one way is where they interact with the ATPase, inhibit activity, that leads to the changes in the cytosolic composition, cation composition, and this leads, of course, to changes in calcium, and this is where we have calcium inotropic effect, and this is called ionic pathway. So the next, the other pathway is signaling pathway, where cardiotonic steroids are kind of messenger. They interact with sodium potassium ATPase, which somehow starts different signaling cascades. Here we have a lot of different facets because it depends what kind of isoform of sodium potassium ATPase we are talking about. This was Gustavo who talked about it. It depends where in the cell membrane our complex is found, and also which exactly pathway one induces by this way. So this is, let's say, generally accepted picture of multiple functional consequences of the sodium of the cardiotonic steroids. It does not mention one more possibility which appeared recently with all these omics approaches. The network pharmacological analysis of the Chan-Su effect on hepatocarcinoma, for human hepatocarcinoma, revealed 82 different targets. I was really scared when I saw a list, but I found sodium potassium ATP there. I was a little bit relaxed afterwards, but these compounds, targets could be comprised into 14 different signaling pathways. So maybe we should also have somewhere back in our heads the possibility that there are other targets or other possibilities of interactions with the sodium potassium ATP and apart from sodium potassium ATPase. But I'm going to talk about ligands. So just to summarize what I said in this historical overview, it seems that we have very similar compounds, but some of them are, and this is cardenolides, some of them are used primarily to treat cardiovascular disorders, while other bufadienolides are used in cancer research. If you make a search in PubMed, most of the papers on bufalin and analogs are related to cancer research. So is there anything in the interactions between these two subfamilies with the sodium potassium ATPase which may account for a prevalence of one effect over another effect, and what do we know about it? Actually, Pedro has told us about preference of different compounds to different conformations of the sodium-potassium ATP. So this is not a new thing. So where does this structural information comes from? It comes from the crystal structures. I would first tell about the enzyme. So crystal structures and recently appeared cryo-electron microscopy data were all obtained on sodium-potassium ATPase purified from pig kidney, or bovine kidney, or shark rectal glands. These enzymes are they have a very high degree of homology. They are very active. We have activity of 1800 micromole per milligram per hour. We know how many active molecules we have per milligram, so we have a well-described preparation. In terms of isomeric composition, we have alpha-1, beta-1 associated with either gamma, if we are talking about kidney, or FXYD10, if we are talking about shark. So the structure, general structure, looks like this. We have this box, blue box. It's a lipid membrane, and I'll start with the monochromic units. We have 3 subunits. So violet. This violet subunit, it's a beta subunit, 1 transmembrane domain, and a very big extracellular loop, heavily glycosylated. That's what we heard about yesterday from Leora. Then we have gamma subunit, that's the orange one, 1 short transmembrane helix, and the rest is catalytic alpha subunit, where we have 10 transmembrane domain helices, they're in green, and a large intracellular loop in different colors because it comprises of 3 domains. So this is P domain, phosphorylation, nucleotide binding, and actuated domain. So hydrolytic site must be somewhere here out of definition of these domains. So this is the general structure of the P-type of the sodium-potassium ATPase. Now, if we want to have to get an information about general principles of binding, it would be nice to have the same conformation of the enzyme in complex with different compounds. And that's what we got long time ago. We got a structure with digoxin, which is heavy, cardiotonic, cardenolide, heavily glycosylated, wabain, cardenolide with only 1 sugar, and bufalin, bufadienolide with no sugar. As you can see, so recently it was also supplemented by structure, X-ray structure of digitoxin that's similar to digoxin, just missing hydroxyl group in position 12, antrayl wabain, which is fluorescent analog of wabain, which we have fluorescent label on sugar here, and 2 very, very strange compound called istaroxime and rostafuroxime. And actually, they are not real cardiotonic steroids or classical cardiotonic steroids because they don't have lacton ring. They don't have lacton ring. For example, rostafuroxime has penta. Okay, and it's that's this one. It's cyclopentadiene, right? Yeah. And the istaroxime, it's even more problematic because instead of lacton, it has ketone group here. It does not have this important hydroxyl group in position 14. And instead of sugar moiety, it has a tail, which is, amino alkyl oxime, right? Or something, yeah. So it's a different, very different molecules. Why they are involved in all this business? Because they are under clinical trials, they can inhibit sodium potassium ATPase, and istaroxime can actually, they say, activate calcium ATPase in the SERCA2 in heart. So it's very fortunate, inhibit sodium potassium ATPase, activate calcium ATPase, very fortunate for their cardiovascular problems. Yes. So now, at that moment, I would like to stress that one should be a little bit more skeptical about crystal structures. A little bit more skeptical about when interpret crystal structures. For example, you can read, and the only way to find out what's right and wrong is actually functional studies. For example, we know from the kinetics that, and this is no doubt, that binding of cardiotonic steroids is induces a conformation transition in the enzyme or in the binding site. So this sentence in the Kanai paper where he says binding of CTS does not require, nor does it cause large conformational transitions. Now we look in the same paper, for example, this figure here, and we compare blue and pink structures. They are with antrailoabine, with the yellow structure, which is without antrailoabine. And I think there is a difference. There are some shifts in the position of the helices. Now, the same thing, we look at the coordination of magnesium ion without antrailoabine and with antrailoabine. And it's also quite clear for me that the coordination pattern is different and there is a change in the position of this M4, transmembrane helix 4. So it's a question of, now it becomes a question of what you understand by large transition or just transition. Another example comes here, again from the same paper where we talk about structures obtained by X-ray crystallography and cryo-electron microscopy. The same conformation of the enzyme E2P from PI, both places. But istaroxime, the ligand, actually has an opposite, orientation. It turns upside down. Here you have, so this tail is facing outside. Here this tail is turned into the membrane. So one explanation, I would say vague, could be that we have just different enzymes, because this is kidney and this is shark. But we talked about very high homology between the isoforms. Another explanation is that the crystal contacts here squeeze the site and does not allow istaroxime to go so deep in as in the other structure. And that's what, but the consequence is that the crystal contacts can change somehow fine structural details in the binding site, right? So I would tell afterwards only about major principles we all agree about and principles which are confirmed by functional studies. So if we put, if we'll make an overlay of these three structures with wabine, digoxin, and bufalin, we can see that they are bound in the same way. That the binding pattern is virtually the same. They have all lacton ring deep in the site, in the almost in the cation binding site, and the glycosylation part is facing up towards the extracellular space. They have this hydrophobic surface directed towards M5, 5, and 6, and 3. and there are hydrogen bonding between the ligands and M1 and M2. Buffalin binds a little bit more deep in the site. You can see it here, it's this pink color, but probably that's because of its hydrophobicity. So now the structure was nice because it could explain us well-known, well-described problem of cation CTS interactions, the antagonism between potassium and wabain binding. And it's known in the clinic that that hypokalemia results in increased toxicity of the digoxin. And very often diuretics which induce hypokalemia are taken together with digoxin when treating cardiac problems. If we now translate it into our language, into a tube, lab tube, that would mean that we can reach full inhibition of the sodium-potassium ATPase at lower concentrations of digoxin or wabain if we don't have potassium. So we have this curve which is more shifted to the left. In the presence of potassium, affinity drops and the curve shifts to the right. So high potassium prevents binding, low potassium or no potassium improves binding. This potassium effect can be compensated by the presence of magnesium, and you can see that this very right curve is shifted to the left in the presence of 10 millimolar magnesium. How do we explain this? It's explained very nicely by our structure where we see that potassium, this is this gray sphere in the binding site, tries to get occluded, which means that it drags part of this M4 helix in the binding site, and in this way pushes wabain out. So it's just the mechanical interactions. Wabain has less space, is pushed out of the site, and affinity drops. Now, is it valid, this antagonism, is it valid for all cardiotonic steroids? And apparently not, because our experiments, Pedro showed this, our experiments with buffalin show that it's surprisingly insensitive to buffalin. It's opposite, actually. It improves affinity and makes this binding more homogeneous. Now I explain what we do when we measure this inhibition curves, because it's probably different from what other people do. We first make a complex. We pre-incubate sodium-potassium ATPase with phosphate and magnesium for 4 hours. So the question of time is solved for 4 hours. And then we take a very small portion of this enzyme and put it into a media, hydrolytic media, which means that we dilute phosphate, dilute cardiotonic steroid, but we know that the complex is so tight that during these 2 minutes we measure activity, nothing happens. It does not dissociate. Remember that we have a very, very, very high activity of the enzyme. So it's enough, 2 minutes is enough for us to measure enough phosphate to determine activity. So what we see here, that in the presence of potassium, we reach full inactivation of the enzyme. All complexes which are formed are stable, very stable. In the absence of potassium, we have a stable complex, and then we have about 20% of something which still has its activity, which means that this buffalin in one moment dissociated, right? And enzyme was active again. So we have a problem, 2 problems. Why is it not sensitive for potassium, and why do we have this heterogeneity? So crystal structure, it shows that, well, yes, we have potassium here that's now in in this green color. Potassium ions, they are slightly shifted in comparison with potassium in the Wabine binding structure. And here we have bufalin, which actually binds deeper, approximately 1.5 angstrom deeper in the site. And it seems that it interacts directly with the cation in the binding site too. So it's electrostatic interactions which stabilizes bufalin. Okay, so is it a property, this insensitivity to potassium, is it a property of the sex-membered lacton ring? So we decided to extend our studies and added cinabufagin. So it's bufalin with a bulky substituent in the ring, D, in the D ring. And we did the same experiments, inhibition in the presence of potassium, inhibition in the absence of potassium, plus inhibition in the presence of other ions. And next table has a lot of numbers, but what we should be aware of that presence of potassium makes, for both bufalin and cinabufagin, makes inhibition complete, which means that we have homogeneous preparation of the complexes. Another thing, how affinity is affected. For potassium, we said that it's probably improved. It's higher affinity, or maybe the same, doesn't matter, but at least it's not worse. For cinabufagin, surprisingly, it is getting worse. So there is a certain antagonism between cinabufagin and potassium, like cardenolides, other cardenolides. It's bufadienolide, right? So we try to understand this, and we turn to molecular dynamic simulation, where again, we took our structure with bufalin, and remember that structure is frozen, it's a snapshot. So we took our structure and allowed all the components to move. And what we saw that there are several binding modes for bufalin. It can actually turn around in the site approximately 180 degrees and expose this beta surface to a different transmembrane helices here. Two of these binding modes were stable. The fourth one was very unstable. And this is in the presence of potassium. Now, if we remove potassium, what we see here that we removed potassium we have here we do not have it. What happens with bufalin? It feels uncomfortable. It feels uncomfortable and either completely dissociate. You can see here the molecule is on the way out, or binds on very high up in the binding site, which in other words shows us that there are different complexes with different stabilities. What about cinabufagin? Cinabufagin, remember that it's the one with a bulky substituent. Yes, it gave us only one binding mode similar to bufalin in the presence of potassium, but in the absence of potassium, we got 5 different binding modes. Those ones were similar to those of bufalin. One is these two. It's higher up, so closer to the exit from the binding site. But these 3 on the down row, they are all show that the molecule is bound upside down, which means that the lacton ring is facing outside. Right. Okay, so it seems that the positive charge in the cation binding site is important for orientation of the our ligand in the binding site. Now the question is, how do they interact? So what we do now, we analyze the distance between lacton ring and the cation in the binding site 2 as function of time. And we do it for bufalin and cinnabuphagin, and we do it for potassium, sodium, and magnesium. All three ions which can be involved in this situation. And what we should look at the size of these peaks, because they reflect the probability of finding of the lacton at a certain distance from the cation. And as we can see, bufalin here, it comes close. It comes to a distance of about 2 angstroms, which means that it's at a, it fits with a first coordination shell of potassium. With sodium, it seems that bufalin is higher up. It does not reach the first coordination shell. It should go to the second coordination shell of the ion, and it means that the affinity is and interactions are lower. The same with magnesium. It's even higher. The distance is even higher, right? So it's only potassium who can, only in the presence of potassium, bufalin comes close enough to interact and coordinate the ion. Cinnabuphagin, because of this huge substituent, cannot come close to the first coordination shell. It is always far away, higher in the binding site. This is coordination between cation in the binding site and the ligand. But the cation, when it is in the binding site, it also interacts with a protein, right? It actually spans two, two bunches of helices. One is this very stable, very stable bunch of helices from 5 to 10, which does not move a lot. And the other one is very flexible, very flexible collections of the helices from 1 to 4. So there is a possibility of interactions of the cation in the binding site too with 5 different amino acids or groups, with 5 different groups, because some of them belong to the same amino acid. And as you can see, potassium interacts. You should see at the depth of the color. It's very difficult probably to see, but trust me, potassium spans these 2 bundles all the time by all 6 coordination bonds with all groups. Sodium spans with all 2 bundles, but with a lesser amount of coordination bonds. And magnesium is always connected to, coordinated by only one side, right? So that explains that while our effect of ions, we can add it here, our effect of ions can be summarized in the following way. So for the first, it has an electrostatic attraction, electrostatic interaction with the lacton ring, and on the other side, cations have different abilities to shape the binding site and keep it stable. Potassium, in this sense, is the best. How I'm doing?
5 minutes.
Oh, are you really? Okay. 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 So we go to
leave our lactone ring and we go now to the glycosylation, because that's another part which was very important, and that's what we can see from the kinetics. Kinetics for aglycone and glycosylated compounds is very different. George mentioned that there are some interactions between digoxin, right, and beta subunit. It's true, we can also see this here. And there are some interactions, possibly some hydrogen bonding with a loop of M1, M2, but we don't know how stable these interactions are. And in principle, we do not have a clear-cut explanation why the kinetics is so different. What kind of stability this sugar moieties bring with it. Then we made experiments and we saw that actually affinity is very much dependent on the number of sugar moieties, and it has a bell-shaped form. Each color corresponds to individual aglycone, so it's different cores, and then we have different number of sugars attached. And then we can see that one sugar residue is very nice. It gives the lowest or the highest affinity, the lowest concentration. But if we add more and more, this bulkiness probably prevent from proper binding. And this is done in 2 different buffers just to avoid technical problems. Oh, yeah, technical, yeah, not to be biased, right? So how we have to look at this glycosylation, and here we have three possible cases. One is the size, how big is the size of the binding site for the cardiotonic steroid. We saw the structure, but structure could be biased. The second one, how big is the glycosylation part, and then the third one is what is the stereochemistry and origin of the atom involved in this glycosidic bond. So let's start with the binding site. We, as I said, crystal structure is just a snapshot, so we decided to see what happens in the solution, and we turned to spin resonance, electron spin resonance. So we synthesized specific spin labeled where we have a core, as a steroid core from cinabufagin or bufalin, and then we have a spin label here attached to this core with a help by a arm or tray, with an arm of different lengths. And as you can see here, it means that our binding or our spin label is placed in a different depth in the binding site. This steroid core ensures very high or reasonably high affinity, so we can reach full saturation of our samples. And we make experiments we compare spectra, ESR spectra, in 2 different conditions. One with a high concentration of wabain, which of course would chase all the spin labels from the binding site. Spin labels will be outside in the free space, and that we can see that it is reflected by very sharp lines here in the spectrum. It means that the spin label has no problems. It can move freely or rotate freely in the media. The second one, where the spin label is bound, you can see that here we have a change. Instead of this sharp line, we have a very broad line here, right? And the shape of this line depends on the length of this spacer arm, which means that the mobility, this broad line describes that the spin label is somehow restricted. So mobility is restricted when the spacer arm is short, and it becomes bigger and bigger, mobility, with the longer spacer arms. It's clear because our spin label moves further and further out of this restricted area. So by analyzing the data, we can say that this the site has a reasonable size. It does allow both chain conformational transition of the spin label together with all the attached substituents here, so they are reasonably big, and by a special method and use of heavy water, we know that our binding site is filled with water. So that's what we put here in the list of our main conclusions. Vestibuli leading to a steroid binding site and cation binding site is relatively wild and water-filled. Yes, now I'm talking the last 2 minutes. I would just say a couple of words about the project which we haven't finished yet. Lucy still work on this because she had children and so on. So it's also productive in another way. We were looking at stereochemistry of the atoms involved in the glycosidic bondage, which means that we are looking at the position 3 in the steroid core. If we start from the very beginning, the parent compound, the steroid, the cholesterol, actually can exist in 256, right? Eight, Herald. No, George is busy. Yeah, in a lot of conformation, 256. Two in the 8 potents, doesn't matter. So a lot of conformation, but you can find only one in the nature. Native is only one, and I'm talking about this most interesting position. But people made, of course, they were interested in this state, this position 3, and they made a so-called epicholesterol, which means that this hydroxyl group, instead of facing up, it faces down. And they found out that this molecule can, for the first, it is oriented in the lipid bilayer in another way. And for the second, it does not support function of serotonin receptor, and it doesn't support function of certain channels. We got synthesized this alpha and beta isomers of the bufalin, and to our surprise found out that it had a huge effect on affinity. As you can see here, affinity, if you just move this hydroxyl group from position up, down, from beta to alpha, then you change affinity from nanomolar to micromolar range. And we cannot explain it from the crystal structure. We don't know. So we again turned to molecular dynamics, and we decided to analyze binding of alpha bufalin and beta bufalin, as well as two different glucose bufalin. One is O-glycosylated, another is N-glycosylated. And this is because people who make structure activity relationship prefer to make N-glycosylation, it's easier. And the information, if this N change O from N is important, it was unclear. Some people say that it doesn't matter. Some people say that it's very important. So we don't know. The other thing is this bufalin. Why do we need to analyze alpha and beta bufalin if only beta is found in nature? The problem is that in rat plasma, after oral application of this chansu, they found alpha bufalin in the plasma and some other places. I don't remember where. And they also say that this alpha bufalin is much more cytotoxic than beta analog. Another thing is that very often these stereoisomers, they appear when one incubates a certain substance in a plant cell culture or in microbial culture. As George told us, they have some special enzymes which make stereoisomers. So it's important to check if what's going on. We turned again to our molecular dynamic simulation, as you can see here. And what we found out, I don't know if you can see, there are different colors, right? And hatched bars and plain bars. And they correspond to different isomers and to different molecules. And as you can see, each particular molecule, each particular ligand has its own preferable binding mode. So now we need to translate our interactions into binding, into KD values, into affinities, and then try to figure out what's going on. We haven't done it yet, so I owe you an answer about effect of stereochemistry, about effect of length of the glycosylation bond and the origin, if it's O glycosylation or N glycosylation. My battery is running soon, but you see, I got a message. Battery is running soon. Do you have some kind of remote control? No? You say battery running out soon. Oh, very nice. No, it's okay. I'm finished. I thought it's you who said that the battery is low. Okay, the last is yeah, that's what I thought. So the last point on my list says that very general. The major binding mode is similar to all CTS, although the probability of binding of certain compound in each binding mode depends on the individual structure. That's very general, sentence. That's it. I just need to show people who are involved and, of course, funding agencies and a lot of people. That's where all my colleagues and I am standing now with a lot of questions and not that many answers. Thank you.
Thank you. This is very intriguing talk. Questions?
A lot of questions, but just one.
Yes.
Obviously about the beta subunit. Your simulations, when you do the simulation, the molecular dynamics include beta or not?
No, it's too big. No, we limit ourselves.
But the transmembrane?
No, because no.
This is not so big.
No, but
it interacts with M1 to M4.
No, as you can see, we
I
have
don't
only,
remember.
We have only the helices which.
My question is if the transmembrane domain of beta subunit is close or interacts with the M1 to M4 domains. If not, I understand why you did not include them, but...
Well, it's close. I don't know if it interacts, right? You can see
yes.
No, it's on the other side, actually, right? Gamma
but
is...
I remember from the works of the crystals,
that
there is an interaction with certain M domains, but I don't remember which of them. Okay,
no,
we can check.
It's this pink one, right?
Yes.
Yeah, binding site for WABA or it's here, and this is here.
We don't see them. You have
oh,
to.
I'm sorry. Yes, I forgot. Yes, binding site is somewhere here.
Okay.
Oops. Yeah, that's
That's the sign.
that's him.
Thank you.
I don't think the slide, but thank you. Every time I understand more. So thank you for detailing and for all the work, of course, that goes behind it. I just have a general idea that I wanted to share. By no means I am an expert in traditional Chinese medicine, not at all. But I guess over the years, it rubs off a little bit on me. So with the extract, the toad skin extract, chanshu.
Chansu, yeah.
Chanshu, right?
Apparently, I don't know.
So the fact that it activates so many pathways in the research that I'd like to look at it. I haven't seen that paper, so it looks very interesting. But one thing with the traditional Chinese medicine, it will never ever be one purified compound, in my understanding.
Exactly.
So it's
exactly.
enriched with bufalin.
Yes.
But there's other active compounds that maybe are explaining some of the pathways that are activated.
Exactly.
So that would be my guess. Maybe if that makes us feel a little bit better. But especially they like to have, they believe, if I understand correctly, that there will always be a balance, especially with toxicity. There's going to be other compounds that balance that out. Or it's fascinating. I don't understand it at all, but my immediate thought was like, oh, it's okay because it's not pure.
It's not pure. It's not. I don't know if it's approved by the administration, this China Food and Drug Administration. It must be somehow standardized.
I bet it's safe. I'm pretty sure. I'm sure it's
No,
been
no.
used.
I mean standard.
Oh, the
composition.
standard. Yeah, maybe
Probably.
maybe it's standardized.
Because
That,
it's
I don't know.
encapsulated. It must be.
Yeah.
They have to do something about it.
Yeah.
They just give whatever.
I don't know.
Yeah. Okay. I don't
think
know.
I can answer
I think
more or
also-
less. There are serotoninergic substances, histaminergic substances in Chansu. The extract, the majority of Chansu is not bufadienolite.
No,
No,
but that's what they say.
no. There are many things. There's just like extracts of the whole
Of the
thing, you
skin.
know.
Yeah.
Of course, there are many, but it's not the majority of the extract.
No, but I mean the rest,
Chansu.
yeah, but the rest of things, the rest of the experiment, they were done with bufalin or cinabufagin or something.
Sure.
Yeah, so I do not advocate to.
No, she's just asking about the FDA approved substance. It's like phytomedicine, you know. There are many things inside.
Yeah. I'm not. I do not take it myself.
Neither me.
I remember that sometimes WABAin is also used to inhibit the proton-potassium pump.
Yeah.
And the proton-potassium pump and the sodium-potassium pump are very similar. Do you think that...
HK.
HK?
Yes. Mm-hmm.
So, do you think that is it possible that given the similarity, Cardiotonics steroids binds also in the same way to this proton-potassium pump?
Well, I don't know. If you refer to the co-handling, so they made, I think, six mutations to create a WABAin binding site in HK ATPase. So it's not a HK ATPase per se. It's mutated.
And the other thing is that the original crystal of the sodium potassium ATPase has a cholesterol
Yes.
included.
Yes.
Is it known, its function, or is it important for the
Well, that's
structure or the function of the pump?
well, yes, you have to have cholesterol in the membrane if you want to have sodium potassium ATPase functional. But I guess it's more like properties of the enzyme. Cholesterol, you are right, it's close. It is co-crystallized with the sodium potassium ATPase, but you cannot find out how strong are the interactions, and you cannot extract it. I mean, to crystallize, we solubilize it, so somehow the molecule cholesterol goes together with the rest of the enzyme. So it's reasonably tight interactions. But how important they are, I don't know. I just know that when we make vesicles and we want to have sodium potassium ATPase active, then we need to include cholesterol in a lipid mixture.
More questions?
So I mentioned that we do some modeling, but we're really bad at it. And I assume that's because, like, when we change the sugar, ours are always the digitoxin aglycone with different sugars on there. Whenever we do any modeling, any minimization, it essentially just ejects the ligand.
Mm-hmm.
I'm assuming that's because we're not accurately figuring out how many waters are probably in that active site.
But it's with water filled.
Exactly, but
Yes.
but our model doesn't really have
No,
that,
no,
right?
no.
We don't have those
Yeah,
sort of
yeah,
details.
no.
Does that make sense to you? I mean, if we started putting model waters in, do you think we would, or is that a fishing expedition?
I don't know, but it is, for example, interactions, but this is bufalin, if with magnesium in the binding site. So if we don't have potassium, and this is Kanai's or Toyoshima work, they crystallized sodium potassium ATPase with bufalin and no potassium. We did with potassium, he did without potassium also. And he said that the interactions with magnesium is mediated by water.
Huh.
So I don't know.
Okay.
So it's important for stabilization, but this is bufalin. You are working with cardiotonic steroids, right? If it was cardenolides.
Okay, so thank you. We can continue. Please, Gustavo. Also, a big pleasure to introduce the bishop. Monsignor. Well, I think Dr. Gustavo Blanco don't need an introduction. Everybody knows him here, but it's a great pleasure to get you here. And now you're going to talk about something else. Yes, it's about the new wobble derivatives for blocking the sodium signaling in polycystic kidney disease. Thank you, Gustavo.
Thank you so much, Luis. for the nice introduction. And of course, I don't have any photos, so everybody can mine. But thank you a million to the Pope for bringing me here. And of course, where's the Queen? The Queen, oh, there is the Queen. And Jacqueline, the Prince. Thank you for bringing me here. And the other thing, thank you so much to the students. It has been great these days interacting with you. And also, I owe you big, because thanks to your gift, yesterday in Tiradentes, the UV rays didn't kill. That hat was very, very useful. So this has been so far a great meeting. Let's see what happens with the last one. Oh, microphone. Huh. Oh, now I go. Oh, that's too hard. Great. So it has been great. We have heard a lot about very different topics, mainly a lot of chemistry, especially today, a lot of these DB compounds here, a lot of different effects. We heard about the alpha subunit, all the mutations, beta subunits, how they are important for cell-cell interaction. We even heard about another pump, the calcium pump. And a lot of these effects in the cardio and vascular system. So it has been fantastic. Now I'm going to tell you about something different. It's related to this disease called polycystic kidney disease. So let me introduce you a little bit about some things of this disease. This polycystic kidney disease in general is a family of diseases that are characterized by this formation of cysts that are fluid-filled, and they act as tumors that keep growing in the kidney, compressing the rest of the kidney tissue, and eventually the kidney doesn't function anymore. So it comes in different, the prevalence is around 1 in 500, 1 in 1,000. It affects all ethnic groups, and it comes in different flavors. There are autosomal dominant forms and recessive forms. The recessive forms is lethal, and fetuses that have it, I mean, the babies don't make it. But the autosomal dominant has a very slow progression, and the disease appears when people are 50, 60 years of age. This disease is responsible for about 10% of the end-stage renal insufficiency. So all these patients eventually go to dialysis, and they get into the list and hopefully renal transplantation. So it's a big problem. It's a genetic disease, and it's caused by mutations in 2 proteins known as polycystin 1 and polycystin 2. And the function of these proteins is not totally known. Together they act as a ion-specific channel, but they have a lot of other functions in the cell. And the most important thing is currently there is no treatment available for polycystic kidney disease. And I'm going to refer to it. I'm going to talk about the autosomal dominant form, and I'm going to refer to it from now on as ADPKD, okay? So here you have the cyst, and you can see how they end up covering the whole kidney. And this is the nephron, the functional unit of the kidney, and they can appear in any place of the nephron, but they are much more prevalent in the collecting duct. And the pathophysiology of this has been under a lot of study, and what appears to be the first event is an increase in proliferation of the cells. And the genetics here are very complicated because, as you can see, the cysts are not, I mean, they could be everywhere, but not every nephron is affected. So this has been under a lot of study, and what happens is you need the defect in one of the alleles, but then there is what is called a second hit, and something happens that the second allele gets affected, and there is where the cyst starts forming. So not every single part of the epithelium gets, it's abnormal, right? And what is this second hit? A lot of times it's just problems, little injuries, in the epithelium, and then there is what is called a futile repair. The epithelium tries to repair itself as all other epithelial cells, but it grows too much. And proliferation is the first event, as you can see here, and the cells keep proliferating, forming this like blister, and eventually the blister pinches off, and now it continues growing, not only by growing the cells, but also by increasing transepithelial fluid secretion. If the cells only grew, we will end up with a ball of cells, right? But for the cyst to grow, you have to put fluid inside. So this is how we got first into these. And it's because when there is fluid movement, we have been talking about the sodium pump moving ions, but the ions carrying with themselves water behind, right? So that's why I got very interested in this disease. So here you see that there are genetic factors that are very important, the mutations in the two polycystins, but something was, and you see here the progression of the cyst. So you need a lot of cyst for the kidneys not to stop being functional. And that's why, because we have 1 million nephrons in each kidney, we have 2 kidneys, and as you know, we could donate a kidney and still be fine, right? So it takes time, and that's why the disease appears when we are 50, 60 years of age. Well, hopefully it won't appear to us, right? But what is very interesting, and this was known from a long time, is that there are a lot of mutations in the polycystin that can lead to polycystic kidney disease. But for example, siblings with the same mutation, one may require a kidney transplant very, very quick, and the other one can keep going for a long time. So it was like, how could it be? Well, and the explanation for that is epigenetic factors. And there are factors like, and some of them have been discovered, but the increase in cyclic AMP is one of those factors. And vasopressin is a hormone that acts in the kidney through activation of cyclic AMP production. So high levels of vasopressin have been associated with a higher progression of the disease. So I'm going to show you data today that another factor that is very important is wabin. And of course, here, endogenous wabin. Thank you for discovering it. It plays a role, right? So we, as I said, the first reason that I got into this was like, wow, if these cysts have fluid, the sodium pump may be involved here. And there was a little controversial data when I started doing this, and there was a group in California that showed that in polycystic kidney disease, maybe the sodium-potassium ATPase was mislocalized. And instead of being basolateral, like you see it here, and the fluid moving from the lumen of the tubule into the blood, it could be misplaced here, and then the fluid will go the other way. And then that will form eventually the cyst. Well, that could not be replicated by others. I tried hard to with different antibodies, and it's basolateral, and this is what we found. But then, you know, I keep studying it. I said, well, okay, let's see what are the properties of the pump in ADPKD cells. And when I say ADPKD cells, we have been working with different models, but we have human models, and those are cells taken from the cyst, the lining of the cyst, and you can grow them in culture. And that's one of the things we have been using. And because people have been studying this for years, there are different models to study this disease. So I got, in this case, is one of it's a mouse model. And these are the activation curves of sodium-potassium ATPase activity versus sodium and versus potassium. In green and in red are the normal and the cystic animals, and these are just, it's the enzyme membranes from kidneys. And as you can see, there is no difference in the activation curve. So, and we did ATP too, and the kinetics were all similar until we came across of an inhibition curve for wabin. And when we measure sodium pump activity versus wabine, here in green, you see the typical monophasic response of normal kidneys. But in kidneys from this model, you could see a biphasic curve. There was a component with much higher affinity than the regular alpha-1, which, as we find it, is the main and almost only isoform expressed in the kidney. So we got the same in humans, and these are the human cells I was telling you about. And this is a control, human cells taken from normal kidneys that have not been able to be transplanted. And look, when you do the ADPKD cells, they have this biphasic response. So we were like, wow, being the cells so prone to bind wabine, and also we use a marker. This is a Bodipi wabine. In the normal cells, Bodipi wabine in low nanomolar concentrations, 3 nanomolar actually, you don't see any binding, but in the ADPKD, you see the binding. So those cells have more affinity. So with that in mind, the first thing I thought, and based on the rodent data, and this is work we did a lot, and a lot of people have done, is that if these are the different isoforms of the sodium pump, and look how they respond differently to wabine, right? Alpha-1 is very resistant. You need a lot of wabine to inhibit it, and the other isoforms are progressively more sensitive. So I said, wow, if we got a biphasic curve, probably in ADPKD there is misexpression of isoforms, right? So we did a lot of work. I was all happy. And remember, until now, the only thing we found is just that difference in wabine. And what happened is we did PCR. I'm showing you only the Western blots. There is alpha-1, and there is beta-1, and no other isoform. So, oh my goodness. Until today, still, we have other data that is too preliminary to be shown, but we're trying to find what is making the sodium pump in ADPKD more sensitive to wabine. And I will tell you something. We believe it's protein-protein association in these cells that is kind of messed up, okay? But I don't have data yet to show you that. But what we started doing is going by, okay, wabine had an effect. Let's continue looking at the effect of wabine so on these cells. So here are monolayers in transwell plates of human epithelial cells from ADPKD patients. We starve the cells, then we apply wabine 3 nanomolar, and then we determine cell proliferation. And here you have the normal cells. They proliferate but little during the different concentrations of wabine. And then look at the ADPKD cells. Wabine increases the growth of the cells as the typical effect of wabine as a growth hormone, right? Basically. So this has been described in a lot of tissues, but the importance here is what? Remember I told you that one of the main hallmarks is first proliferation. So proliferation, if it's increased, that will increase cysts, right? So then this is a factor that is important. And this is the same result, except that we counted the cells instead of using the MTT assay that also measures metabolism. And then we measure the mitotic index, and basically all is indicating that these cells grow a lot more than the normal cells and the ones without wabine. So, and we did a series of studies following the Toledo and then the Marshall group, and we found out that the effects of wabine could be, see, you put wabine, the cells increase and proliferate more, and then if you use inhibitors of the EGFR, SARC, or MEK kinase inhibitors, you abrogate the effect. And here is the phosphorylation of ERK. Nothing happens with wabine in normal cells, but in the ADPKD cells, there is a huge increase that is dose-dependent with wabine. If the doses are in the nanomolar range, you get proliferation, you get the activation of the signaling pathway, the sodium pump signalosome, as Jen used to call it. But if you start increasing wabine, then there is toxicity, and obviously the cells stop growing, right? So here you can see the PERK levels, and again, with different inhibitors, you abrogate the effect. And the interesting thing is that, when there is more proliferation, obviously, all the cycling-dependent kinases are involved in cell cycle, right, and proliferation. So when you look at, for example, we look at a bunch of them, but here I'm bringing you P21, and these are cycling kinase inhibitors. And because, look, with wabine, they become decreased, which means then that the cell cycle can proceed and the cells can proliferate more. So the effects eventually of wabine in the cells are happening through this cascade, and eventually the cycling kinase inhibitors somehow are activating, are involved in gene regulation and in nuclear effects in the cells. So the other thing that we found, and I'm going to go very, very quick here, is that I'm going to just describe this first thing. We look for also apoptosis, because in ADPKD, it has been shown that apoptosis was increased. And it's true, wabine increases apoptosis. I'm just going to show you this, but not necrosis in the cells. And we know now that after studying all the pathway, that this is happening because of activation of wabine of the intrinsic pathway for apoptosis. But then there is more growth, there is more apoptosis, but obviously the growth prevails, and then the scale gets shifted to that side, and because proliferation is more, that is the outcome we see with the cells. But look at this. This was very interesting too. If you grow the cells in monolayers, and then you treat them with wabine, and then days later you measure the amount of fluid that is moved in these cells, you can see that, in the presence of wabine alone, not much happens. This is something that was described. If you apply for scoling or dibutyryl cyclic AMP, remember I told you cyclic AMP is a factor that is important. Secretion in ADPKD cells increases, while in the normal cells is very low. Why? Because the normal epithelium normally reabsorbs and doesn't secrete, right? So, but look, if you add wabine and forskolin, there is a synergistic effect. So wabine is important for cell proliferation in ADPKD, but not only that, also for a fluid secretion. So those are the two hallmarks of a cyst to grow. So definitely these were the first indications that wabine has pro-cystogenic effects in ADPKD. And this was in monolayers, but then you can grow the human cells in collagen matrices and they form cysts. And these cysts are bigger when you use wabine in combination with forskolin. Maybe you can see here the cyst a little bit. These are micrographs. So we also checked these in whole mice, in whole kidneys that were embryonic kidneys that we can grow in culture. And if you apply wabine, and in this case, 8-bromocyclic AMP, you get a synergistic effect with formation of cysts that you can quantify. Look at the kidneys here, and you can see these last ones. They have a lot of white kind of blisters there. Those are the cysts. And we can measure the area of the cyst and determine and quantify this as it's quantified there. And we explore the pathway, and it turns out that the pathway is the same, activating, the MEK-ERK pathway. So the other thing we found out is that this was a debate in the ADPKD field for a while, and also that was because people thought, oh, the sodium pump is mislocalized. The fluid going into the cyst may have sodium preferentially, right? That's what you would think. Well, there were a lot of debates, and there is sodium, but there is a lot of chloride. And it turns out that the CFTR, the cystic fibrosis transmembrane regulator, is very important. It's located in the apical membrane of the collecting duct cells. It's very active and causing movement of chloride that is followed with water and sodium, And that's the main generator of the secretion. So, but look here. So we grew the cells and we look for secretion as you can see here. And when you add both wabine and forskolin, it goes up. But if you inhibit the CFTR, it goes down. So it looks like the effects of wabine are mediated through the CFTR, which is the cause of the mechanism by which cysts progress. And we did the same in microcyst, and you see the same effect. And we also explored this using a mouse model in which CFTR is not present. We work with heterozygous mice, and then it's in embryonic kidneys. And if the CFTR is not present, wabine, even forskolin, do not work. So that and then we did experiments in the human cells, and this is short circuit current, and you can see here without wabine, and when you apply wabine, look, the short circuit current increases, which is dependent on the movement of chloride into the cells. And you can inhibit these if you use the CFTR inhibitor. Here you see the full curves during time. These are untreated cells treated only with wabine, almost no increase. If you apply forskolin, look, but if you apply forskolin and wabine, you have this big effect. So, and the reason that how wabine activates CFTR, well, all we know for now is that the amounts of CFTR at the plasma membrane, look, this is time, 1 day, 2 days, the CFTR increases at the plasma membrane. We have done this through biotinylation assays and pulling it down, and the amounts increase. This is another transporter that is not changing, and it turns out that CFTR becomes phosphorylated, and basically we end up finding that we don't know exactly what happens after the activation of the typical signalosome, but between that and CFTR, the activation is through increasing the levels of CFTR at the membrane. Apparently, we have other studies showing that the reason there is more CFTR is not because it's synthesized a lot more, it's just not internalized. So then wabine can activate that too. And this is just following what Jen did and Sandrine and all the group there, is that definitely in these cells, the sodium pump is the receptor for wabine, and it's distributed, and here is a C line. You can see it very well. It's basolateral, but once you apply wabine, some of the alpha-1 subunit, in this case, the one we measure, not disappears, but is reduced at the plasma membrane because of internalization of the receptor after binding with wabine. But with that said, it's slow, it's minimal, I mean, not very high, but when you measure rubidium uptake as a marker for potassium, when you treat the cells with wabine, there is less rubidium uptake. So that means that not only, and the intracellular sodium compared to normal here goes up. So that means that there is also some inhibition of pumping. So now the mechanism is through signaling, but also through the pumping that gets somehow inhibited a little, maybe by wabine or by the internalization itself. And this is the general mechanism. So far so good, students. Is it okay? Okay. You tell me, you stop me if there is something. And then look, another property of these cells in ADPKD, people have found that they are de-differentiated. And they are not they are epithelial cells, but they start losing polarity, and especially not only some of the polarity, most of it, the planar polarity. So they don't form this nice monolayer anymore. So we check without and with wabine here the effects on E-cadherin, which is a marker for epithelial cells, and look, it goes down. N-cadherin instead, that is more for fibroblast, and it goes up. And then we look at a series of markers, alpha, SMA, collagen go up, but then others like vimentin, fibronectin don't change. So, and then the normal cells, there is no effect. So the conclusion here is that and this happens through the TGF pathway. I'm not going to use too much time here, but the idea is that these cells become into a meta-stable state, and people know this as partial EMT, and we have been discussing this with Luis. He's doing very nice studies with bufalin in this respect in MDCK cells. Yes, LLCPK cells. So yes, these cells become a little undifferentiated, and those are parts, are all components, proliferation, secretion, and different components of the ADPKD phenotype, which are all promoted by WABA. So we also saw some effect on cell-cell adhesion, but I'm going to show some of these. Let's go. And for example, we treat with WABA and let the cells go for different times in suspension to form aggregates. And look, they form less aggregates when you have WABA, which means that they cannot maintain their cell-cell junctions that well. And they are more mobile. If you measure migration of the cells, this is just doing a scratch assay. You basically plate the cells, and then with a pipette tip, you just make a scratch, and then you follow how they grow and repair the epithelium. And look, these are the normal cells. You kind of see they're trying, but instead look at the gap. It's a lot smaller. It's because the cells are moving in and repairing the epithelium because they're proliferating more when you have WABA. So, and the interesting thing is somehow the cells manage, and we don't understand all the mechanisms here. You apply WABA, they become more mobile, more undifferentiated, but they are still not leaky. They maintain their permeability, and we have used for these dextrans, and look how they move in transwell plates from the bottom on top or vice versa, and we have used dextrans of different sizes, and somehow these cells manage to do that, which I cannot explain it fully, but it makes sense. Because if you have to make a cyst and you keep putting fluid, and if they are going to be leaky, the balloon is going to ooh, right? So somehow, and these are a lot of people in my university. It's Alan Yu. I think you guys know him. The Mexico group knows him well, but he wears some clothing and he's studying this, and he's studying this in ADPKD, and still not very clear what's going on. So this is a summary of all the effects of WABA and some others that I have not shown you, but basically nanomolar concentration, it enhances proliferation, it increases fluid secretion, all is mediated through the EGFR, SARC, MECAR pathway. There are EMT changes and relaxation of some of the cell adhesion molecules, but so WABA then is a big pro-cystogenic, let's say, factor that contributes to polycystic kidney disease. So, but all this is very nice, but it's all in vitro. So the moment came to start doing something in vivo. So, and for that, we started using different animal models. So the first one we use is this is a very known model of ADPKD. It's called the RCRC because mutations in these amino acids. And polycystic, this is in polycystin 1. It's an 11 transmembrane domain, big molecule. And the advantage of this model is it has very slow progression, like in the human. The disadvantage for whoever works in the lab is you have to wait a lot for the data and the results. But anyway, so the first thing, this is just to characterize the model, this model, but like it worked like other models. This is the wild type, a WABA inhibition curve, monophasic, and look at here, biphasic. And then when we look at the sodium pump, I don't know if you can see it well, but take my word for it, it's basolateral. So it's kind of normal in terms of that. But then and this is all the work by Jordan Trant in our lab, she started injecting mice with wabine, and she starts at day 7, because that has been shown that for epigenetic factors is a window between 7 and 30 days, and then you can continue from there. And look, she had been injecting daily wabine to maintain physiological high doses of wabine for 1 month, 2 months, 3 months, 4 months, 5 months. And then look, these are the wild type kidneys. This is the wild type plus wabine. You don't see much difference. This is the RC without wabine. And look, when you add wabine, do you see the white areas, the spots? So we can quantify this in the sections, and those are the cysts. And the kidneys get bigger, right? So here is the quantification of that. And as you can see here, well, the wild type is down here. And basically, look here, the RC without wabine and with wabine, the growth of the cyst is much more. Not only and this is measured through the cyst area and using just software, but also the number of cysts is increased. And the interesting thing is you plot cyst area versus cyst number. You know, when you add wabine, it goes down, which means that more new cysts grow. And there is an increase in number of cysts that is higher than the increase in the size of the pre-existing cyst. So that was the first indication that in vivo, you know, wabine also has effects. And also this is just, we look at another parameter, fibrosis. And fibrosis with picrocyrus red, a marker for it, and it's also increased when you use wabine in the RC mice. So the other model that we use, because our reviewers of our grants have been super critical, and for you will laugh, but it's hard for a lot of us to convince them that convince other people, and we have to work together on this. We have been talking with Sandrine a lot about this. They were like, oh yeah, fine, wabine is doing this. How you know it? Through the sodium pump. And that's the typical question they hit us with. So the first thing we tried to do, we made a mouse in which you can decrease the levels of alpha-1. It's a flux mouse, and Sandrine had very good results on that. She showed them. But this is just looking at the kidney. And the problem that we have there is that we could only reduce the sodium pump about 30%. We could not, it's good, but it's not, we wanted like, you know, a lot. But look at, when you do that and you measure the cyst area, in mice that are just the RC, and this is a decrease in alpha-1 in the background of the polycystic kidney disease model, right? And there are less cysts. So if you have less sodium pump, then you have less effects of wabine. That means that the sodium pump, I know this sounds like ridiculous to tell you this, but the sodium pump is the receptor of wabine. So anyways, that, you know, it was, to us, it was kind of obvious, but it works. And this goes back to all the studies that Jen has done with LLCPK cells, showing that, you know, the pump is always pumping. If you start decreasing the levels, the first thing that stops is I'm not going to signal anymore. I have to pump only, right? So this kind of goes with that. And actually, but there are other things we learned from this model. That, you know, we have signaling, and I told you there is a little decrease in pumping, but it looks like the signaling is what prevails in the effects of procystogenic effects of wabine. So the other model that we did, just to keep convincing ourselves, is go to that side, mutate the amino acids there, and basically what we did is use the Lingrel mouse. That's the wabine-sensitive mouse. Remember in the intro, I showed you that 2 amino acids there are critical. We switch them, and we make now look at this, the ADPKD mouse that was biphasic now is monophasic, but the binding constant here is that of the high affinity component only. So all the pumping, this is a global knockout, in this animal is wabine sensitive. When Jordan injected these mice, she noticed that there's a tremendous growth of cysts, a lot more than in just the RC phenotype. And the important thing is that the ones without wabine, endogenous wabine injected also is higher, which that's telling us, John, that endogenous wabine must exist. And of course, of course, I can put a seal on it too. So fibrosis is also worse. So the increase in the wabine sensitivity in this other model also affects, it's another, maybe not direct, but indirect way to show that wabine effects are through the pump, and the wabine affinity is important. And then the other thing we focus on, and we started with the cells in culture. Oh, something is not showing here. Well, so we looked at the importance of caveolae in these. And just to make the story short here, what we use is like a bomb, right? We use methyl beta cyclodextrin, and when you do that, the effects of wabine in cell proliferation or in ERK phosphorylation are abrogated, right? Because you disrupt all the caveolae and let's say the site where signaling starts, right? So we decided to use the caveolin 1 knockout mouse and cross it with the PKD mouse to now see what is the effect of not having caveolae under the PKD background. And this is just the PCR showing that caveolin 1 is not present in this mouse, but it is in the ADPKD. This is just to show caveolin is not there, and sodium pump is basolateral. And we did electron microscopy, and you see some caveolae there. Maybe you can see them, but in the caveolin 1 knockout, and we look at a lot of fields here, it's not present. So there is no caveolae. So in this mouse model, this is the control, or our ADPKD mouse. You inject wabine, and you see the increase in cyst area. But if you don't have caveolae, look, there is no response. And even it's going down more. And see, look, you can see it here. You can compare. And this is using a marker, Ki67, for cell proliferation. See, the proliferation is gone in vivo. So caveolae, we didn't discover anything very important, but caveolae is very, very important for signaling and for fibrosis there. But look, the other thing is, and this is what we are after now, is the biphasic curve of the RC mouse disappear, becoming monophasic. So there is something in the caveolae. Now we are getting closer to try to find out what is in there changing the wabine affinity of the pumping ADPKD. So, how am I okay for a few more minutes? Okay, yeah, don't sleep. See, this is the problem when you are the only one between all the research and the party. Yeah, that's an issue. So, working with a collaborator in Minnesota, we started, she started modifying the structure of wabine and make a bunch of synthetic wabine derivatives. And in this case, most of the change, we are not working with the sugars. So, George, we need to talk. but what we decided to do, and for this, I'm not going to get into all these, but we had another project and we found that the sugars have effect, but it wasn't too drastic. And then because the medicinal chemist that is working with me said, oh, less steps, and we work with these other two groups, the lactone ring and the backbone, right? And she made triazole analogs in C17, nitrile analogs, and then substitution in C3. And some of these compounds, you see here one of them, we call it compound 25. We did the docking and it binds very similar to wabine, but actually it gets kind of, it binds, I don't know how to explain it, but this is a little embarrassing in front of Natalia, but it's tighter, the binding. Not very scientific word. Okay, that's fine. Let me go into physiology, which is what I know a little more. So here we have a bunch of compounds. We tested them against cell proliferation, and we used these at a concentration of 10 nanomolar. and the first, and this is in the absence or presence of wabine. And you can see here that some compounds, and in blue I pointed some, there is not an increase in cell proliferation. See, some of them had no effect. You add wabine, this is the control, proliferation increases, you add the compound, they did nothing. But some other ones reduce it, and reduce it to control levels. Some reduce it too much. So we said like, that must be toxic, right? That was the assumption. So we picked some compounds that had that property. We checked for their effects in p-ERK, and you can see here ERK phosphorylation. And when you use a couple compounds here, the phosphorylation is inhibited. So these compounds have, the ones that we picked, 25, it has relatively good solubility and permeability. We have done some basic studies, some very basic toxicity. This is an anti-proliferation assay. Look, wabine in high concentrations basically kill the cells in culture. Instead, these are a couple compounds here that show that they are not tremendously toxic at least. And this is an assay called the HERG assay. HERG is a channel that is expressed in the heart, and this tells you a lot about heart toxicity and toxicity. It's used as a kind of gold standard for toxicity in pharmacology. And again, this is wabine, our compounds are here, and these are other controls. So the compounds have at least seem not to be too toxic, right? And they have very good metabolic stability. This is in like 5 different species, and this is using hepatic microsomes and letting them chew up the compound basically and degrade it, and you can see a lot of the compound is left, so they are stable. So with this basic information, we also have done some pharmacokinetics, and in the case of compound 25, after injecting it, following it in plasma, we have some data on some tissues, but I didn't bring that. But you can see that, this is up to 50 hours. The levels are they drop, but they are kind of relatively maintained. But then the important thing is we went to the RC mice, and we injected them either with wabine, and the cyst area grows. We injected them with one of the compounds, and it drops. And with the compound and wabine, it drops too. So this compound is kind of, and the same thing with this other compound, basically same results. So this is a preliminary data indicating that these compounds may work in vivo. So we're excited about these. And I'm going to finish there and just give you but the main conclusion is, all the cystogenic effects of wabine, maybe we can prevent using this approach and these compounds. Still, we need to know a lot more about mechanism, because these compounds are and I have some data for that, they are binding to the sodium pump, but somehow they are not allowing signaling and maybe acting as competitive inhibitors of wabine, right? That's kind of I'm saying we have to do a lot more, okay? But that's kind of where we're going. Okay, and with this comes the acknowledgements. A lot of people in my lab has been working on this. The main was, well, Jordan, and then Jeff in the lab has done a lot. Gladys has done a lot. And these two are students. He did all the CFTR studies, Kyle Johnson and Anna. And when and this is my collaborator in Minnesota, Gunda Georg. And we have, thanks to NIH for funding, and also thanks for making us suffer so much with this project. No, I shouldn't have said that. All right, and I will stop there. Obrigado. And if you have questions.
Thank you very much. A lot of data, interesting data. I have one question.
Yes.
Have you tried rostafloxacin?
No, I have not. Actually, I had a reviewer that told me
did you? You said that it's an inhibitor of the activity. You said that of the activity of the activity, of Wabine? Is an inhibitor of Wabine or antagonist of Wabine? No.
Rostafuroxine?
No, your compounds.
Yeah, when they bind, on the activity.
Yeah.
Yes, right. We have to do more studies to really tell you, but yes, they bind to the, especially the high affinity component when you use it at low doses. It's interesting. At low doses, it doesn't affect the, I may have the data, I can show you later. It doesn't affect the sodium pump from membranes of normal kidneys, but then it does inhibit the high affinity component of the ADPKD cells.
Fascinating talk as usual.
Oh,
right?
well,
With all
thank
the
you.
theater and drama and energy. So, follow on to rostafuroxine. In a lot of the paradigms where we turn on SARC with Wabine, low doses, digoxin will block it. Switches off the signaling completely.
I remember Mordy telling me that.
No, it works beautifully. So, so the obvious question is,
use it.
Did you try DIG in your system?
I did not. Yeah, that's a very good.
It's simple. It's cheap too.
I know.
Does patients have high endogenous Wabine or just are more sensitive to the normal Wabine?
So all I can say, we tried it, but the assay really didn't work for us. And what didn't work for us is the preparation of it. So I need to talk to John Mora. I sent a couple emails long ago to Manunta, and we tried to reproduce these and Bagrov, and we couldn't get the preparation right for the assay, really. So in our hands, it has been a nightmare, basically. So, but there is a paper by Bagrov. He showed 11 patients with in end-stage renal disease, and 2 of them had polycystic kidney disease, and showed that Wabine levels were not increasing blood. But when you think about it, in our case, the levels don't matter. It's what matters is that there is this high affinity for it. So that's why I left it there. But a lot of people is asking me, and we are not good at measuring it. That's the problem. It would be nice. We have samples. We have access to samples.
And what is the reason for the low affinity? Does the pump change or it associates?
You mean for the high affinity? Yeah. Well, we don't know for sure. Definitely, it's not based on a change in isoform, let's say. But we believe it's some association with proteins in the ADPKD cells that is messed up. And we are doing proteomics and all that, but I don't know yet. Yes, we are isolating caveoli. That's what we're doing. Yes. So and all these branch off because you knock out caveolin and there is no effect, and the Wabine response is different, right? So, yeah.
So I met a lady in Southern California, and she's got a family history of
ADPKD.
Yep. And one of her sisters has both kidneys affected. She's on dialysis now.
Yeah.
The other sister doesn't have it at all.
Yep.
And she has one kidney that's affected and the other is perfectly normal.
Yep.
Isn't that amazing?
It's
so when I
amazing.
talked to the people at our place, they'd never heard of that. They'd never seen a patient where one of the kidneys was completely cyst-ridden and the other one was perfectly normal.
So,
do you have any idea about why that might be?
Well, there is a theory for this. Let me start by saying that I'm not surprised about it because because for this reason, there was somebody that passed away now called Jared Grantham. He was actually the one that originally kind of studied the pathophysiology of this, and he was in Kansas, and he funded the Kidney Institute, and our institute in the university is one of the centers of, and that's why kidneys come to our, and I when I go to their seminars, a lot of times the clinicians are showing this, and one kidney looks like perfect, and the other one you see all these cysts. But there are different things here, because the problem with ADPKD is the diagnosis. You know, normally you go to the doctor and they measure kidney function, GFR, right? Glomerular filtration rate. And the problem is the glomerular filtration rate, if you remember the figure I showed, you know, with that line, basically it's maintained for a long time. And suddenly when the cyst really reach a critical size, is when the functions start declining fast. It takes 50, 60 years, but the cyst appear when the baby's in uterus, but they are microcysts. And the problem is when you see these normal kidneys, unless you go and do, and you're not going to, you know, in a person, you cannot take the kidney to see this. There are a lot of microcysts. So you may say, oh, it's perfect. And then you miss it. They are still not good parameters to clinically diagnose and especially categorize the disease for progression. That has been a problem in the field. But I'm not surprised of what you're saying. But she may have microcysts in the kidney, but then if she's an adult, that's not going to affect, that sibling is going to be fine. But the mechanisms are not known. The genetics are complicated. There are about 2,000 mutations, including truncations and other things that can cause PKD. But then you need the second hit. That other factor has to affect the other allele, and which are those factors is not that clear.
So we all have cysts, though, don't we?
It's a frequent, yes. Some it's not rare to find in normal people, but few, and especially when people age. But they are hard, yeah. They are at that point they are not critical, right? So, but yes, it's. What was
Activate.
Well, forskolling is an activator of cyclic AMP and then look, I don't know if is there another minute? Is that okay? If not, we can continue later. But there is no treatment for ADPKD. But what Jared Grantham found is that cyclic AMP levels are elevated, and vasopressin is a hormone that is basically constantly hitting our kidneys. I mean, that's the one that tells our kidney you need to pee more or less and control our water balance, right? So, and it works through cyclic AMP. So the levels of cyclic AMP in the kidney are relatively elevated compared to other cells. If you go a little higher, and I think that's the effect of guanine that needs forced secretion for scolding, and they are additive. You need really that extra push for more cyclic AMP, right? So cyclic AMP is a factor. Because of that, one of the only therapy that is out there, but it's not good. So that's what I said, there's no treatment. But Grantham came with the idea of like, why don't we inhibit vasopressin? And tolvaptan is a drug for that. But then you create diabetes insipidus. And these patients, you cannot tolerate that because they're basically constantly drinking in the bathroom. And everybody drops the clinical trials. They were very hard to do because people are like, no, I'm done. And that's the thing. So cyclic AMP is the critical thing.
Okay. Thank you very much.
Let's party.
Well, first we have the flash talk ceremony.
So I ask you for all people that presented the flash talk came here, please. Maria Eduarda, Eric, Ítalo, Carol. So first of all, I want to applause for all the people that make it. We have a lot of undergrad. English is not the first language, so we are proud of you to take I want to talk. accept the challenge. So only for that, you are all champions, okay? But the committee, of course, it make the evaluations. It was very, very hard. So they decided to give you like 3 awards. Two is outstanding, and one like an honor mention. Okay, and thanks Dr. Blanco and Dr. Hamelin, they generous make a donation for have some money for you guys for the honors. So, the first one that I want to call for a honor mention is Maria Eduardo Botelho. Congratulations. So, for the outstanding, have two. Eduarda e Venise. Congratulations. And the other outstanding, Ítalo Leonardo Diogo. So after, I'm gonna take the information for make the pics for you guys. And congratulations all. So just a few announcements. So 7 o'clock is a party in Libertaste. So gonna have music, food, beer. So gonna be great. For people that have to travel tomorrow for the airport, gonna be 10 o'clock at the hotel. And for the students, the bus is 9 o'clock here in the campus. Okay? So you finish. As a host.
Well, so I would like to thank you all, to thank the international speakers, the national speakers, and the students, all the students. You're great. So thank you very much. And congratulations for the Flash Talk presenters that received the certificate and that not received. So they are all great. And I guess I can finish this saying that maybe we can see in the next year or 2 years from now in the 90th meeting on cardiotonic steroids and the sodium pump. Thank you very much for all of you. Applause now, applause. And maybe we can take a picture of everybody here.